to do the honors.
Good morning, everybody, for the first session of today. This is session 8, titled Translating Evidence into Practice. And the first topic of today is the evolution of pediatric clinical trials, looking beyond survival statistics. I would like to welcome our speaker, Mark Rello, to give us this talk.
Morning. I'm Mark Rello. I'm from the University of Southern California, the Department of Population and Public Health Sciences. I work at the Norris Cancer Center, and I'm an emeritus member of Children's Oncology Group. Oops, one back. I have a disclosure. I work on data monitoring committees for Merck, Sharp, and Dome, mostly in adult oncology. My outline today, I'm going to talk about progress in the cure of pediatric cancer and what that means for the way we've always been used to running trials, randomized trials. I'm going to talk how that affects it. Then I'm going to talk about some past uses of historical data in how we design trials, because pediatric cancer is a rare disease, right? In the tens per million. Then I'm going to talk about how we have to change how we think about these data. I'm going to turn you all into Bayesians. We're all going to love that now. Then I'm going to talk about our design approach with historical data and something called commensurate priors, a way we're trying to move to utilize our history in treating childhood cancer. I'm going to talk about the cost. This is not a free thing that we do. And then I'm going to talk about what we as statisticians have to do to make this a better way to analyze data, and what you guys, you holders of the data, what you have to do to help me to do this too, because it's a cooperative affair. We all have things to do in the next while to make this work. So I'm going to show you curves that we all love in children's oncology because it's great, right? This is the survival in ALL. This was the survival when my predecessor, the last the group statistician before me, this is where ALL was when he came. This is where it was when I came, where it says 1983, which is a very long time ago, and where we are today. We are up in many groups over 90% cure, right? So how can you do better though, right? There's not a lot of room left. When we look at AML, it's better, still not great. We are curing over half of children, probably about 65%. And this is my favorite because it's Ewing sarcoma, which I do. And you can see here's where we were about 30 years ago, and now we're up where the blue line is. So we're making really good progress in curing people. But what this leaves us, right, is it leaves us now in our conventional design where we have to randomize the two groups concurrently. It makes it much harder for us to do. And it's because, as a statistician, in order to make progress, you need to observe events. People have to have events. And so what we have now, and I just wanted to show you where we are now for trial design. This is our AALL 1732 trial. It's high risk B-cell ALL. We need almost 4,000 patients to make progress, to make one advance. And it takes us 5 years and 3 years of follow-up. That's a very long time. And for newly diagnosed non-metastatic Ewing sarcoma, another group that I do, it takes us 910 patients randomized to two therapies over 6.5 years and a year of follow-up. It's a long time to make progress to test one thing, right? We gotta keep moving here. I'm going to talk about some ways I think this holds potential for changing how we design trials, who gets involved, and how we can make progress. Now, we have used historical controls in the past. Carlos is going to talk about some trials we did together that used historical data. In COG, we have a vast database of trials going over about 50 years of accumulated data available. We characterize patients, we characterize their outcomes. So there's a data resource we can use. In the NIH, there's something called the Phase 3 Data Repository. If you read a manuscript now from sometime from about 2015 on, if somebody publishes a primary manuscript, those data are there and you can request them. So any data that were there that supported a manuscript table or a statement is all out there for the taking. And the last one I'm going to talk we're going to hear about today is D4CG, Data for the Common Good. Used to be called the Pediatric Data Commons. And there's another set of data, patient-level data, from not only clinical trials, but from registries. And you can get at these data after you've had some scientific review. But these sources are important because they're going to populate how I think we should be doing more trials, using our past history to help us along. All right, so our current approaches to historical controls, the ones we've done in the past, have been kind of clunky. And part of it's just because of the work that's been done on understanding how to incorporate clinical trials. We have done phase 3 studies of ultra-rare disease, nasopharyngeal carcinoma and adrenocortical carcinoma. And what we did was we went to SEER, we went to people's own data set. We created what we thought was a synthetic gold standard. If the therapy was going to be good, it had to be at least as good as a gold standard. And we designed trials to be better than that. But it wasn't real data. It was what we thought from what we got from the data we were able to gather. Now, we've also extended this now in the past 10 years to phase 2 studies where we assemble a cohort of previously treated patients. Usually these are patients who do well. And we take all the concurrent patients we get, we assign them to the experimental therapy. And we use some methods that were developed around 2014 to take into account the way in which data accumulate in the historical controls. And this is something called a frequentist methodology, which I'll make you forget because I'll turn you all into Bayesians. And the thing that these assume no drift in outcome over time. That's why these methods work. And I think that's kind of a dangerous assumption, that we actually don't get better over time just because we have better supportive care. So we need some methods that somehow let us do that. This is a terribly dense slide. Just I like it because it tells me a lot. That we, up until about 5 years ago, we used these top approaches called sequentist and frequentist. And now we're going to talk about some Bayesian methods that allow uncertainty about the historical controls, and we'll figure out some way to quantify that uncertainty. Now, all right, we're all going to learn to be Bayesians. We all read these papers, we see risk relative hazard estimates, we see confidence intervals, but I want to think differently about how we take data in and change our minds about whether we think a therapy is effective. So what I'm going to say is we're going to start off, any trial you have to, if you're going to enroll patients, randomize patients, you have to be at equipoise. You have to believe your new treatment could be just the same as the old. And our general way of thinking it is that if this is no effect here, we are as likely to believe that the treatment is good as treatment is good, it's this way actually, and that the treatment is bad. So I'm going to, as a statistician, quantify our unknownness. Now this is called the diffuse prior because it says our belief is the new therapy could be anywhere up to 22,000 times more helpful or 1/20,000th time as harmful. So it's a pretty diffuse prior. I'm not willing to put a lot of weight on my treatment being better or worse. Pretty diffuse, but I am ruling out very large differences in outcomes. So that's how I'm going to start. And then what I'm going to do is I'm going to gather patients from our model, and then I'm going to modify my prior beliefs with our current data. And I'm going to use a rule that was developed in about 1763. So as statisticians, we think, well, you know, a modern rule, but we like to use it anyway, because it was in the last 1,000 years that this was developed. And this is the only equation. You will not be tested on it later. this is how we do that recalculation. And it's called getting the posterior distribution. We had a prior belief, we get data. Now we're going to have a posterior belief. And if our revised thought of how effective the experiment is, then we are, if it's sufficiently large, we're going to adopt that new therapy. All right, and I'm going to show an example here. So this is an example from how we designed the trial. This, you remember, is our prior belief of what we thought the therapy was like. Could be good, could be bad, but it's centered each way. And this is the outcome of the experiment, right? So I'm not going to go into it, but that outcome actually, if you did a conventional log rank test, you wouldn't reach 0.05. You just wouldn't. Not quite there. But when you combine the new evidence with the prior belief, this is what you get for what we believe the outcome is like. So the area under this curve is the chance that we think the new therapy is better. It's 99%. So we are able to take our prior belief, add it to current data, and that's what we believe. And in that case, I would argue that you have to accept that new therapy. 99%, it's got to be great, right? It's great. Well, let's show you another one here. What if this is what I get for my result? If I look at this, what's my chance that the new therapy is good? It's the area over to this side of the curve. That's 68%. All right. Should we use the new therapy now? More than half, right? But you know, we have an experimental therapy that works. I mean, a standard therapy that works. Why would we move it? Because now we have to do a whole bunch of new stuff to make the new therapy work. So I'm going to argue that we don't quite know what it is. That's part of my job as a statistician, is I'm going to help you understand what is a good cutoff for that probability. Because right now you don't know. And actually, I don't know very much either. So it's a journey we're going to take together. So our decision here is we're going to accept the new regimen if it has a high posterior probability of being better. What does that mean? Well, from COG, we did a study, AHEP 1531. It's an international trial of liver tumors. If the chance that the new therapy was better than 80 was 80% or more, we were willing to take the new therapy. But we also did this a while ago. It's 1999. And our rule then was it has to be greater than 90% probability that it's a good therapy. And that's what we accept. So, and I'm going to talk about one in a moment that's 95%. Why is that good? Why didn't I choose 85%? Well, we'll talk about it. Again, part of my work to convince you. All right. So what I'm going to talk about a design approach that we did to design a new trial. I'm going to go ahead. This was our previous trial of newly diagnosed metastatic Ewing sarcoma. We randomized between two therapies. You could tell we did not hit a home run with these therapies, right? You can't even put a piece of paper between the two curves. We didn't do a great job, so those are pretty equivalent, right? So now we have a new trial. There's 298 patients randomized to that trial. Now we're getting ready to do a new trial, but this is a huge data resource. Why do we want to randomize now just in new patients? So let's talk about how we would do it. We're going to have a design approach where we randomize patients to the current standard of care and a new therapy, but not one-to-one. We're going to exploit by incorporating historical data from that large data set that we had. And so we incorporate these newly documented data, and we're going to decide to accept if the new treatment is better with high probability, if that posterior probability is large. I'll tell you right now, we decided 95%, and I'll give you the scary stuff that we had to go through to do it. We also had a rule that says we will abandon the new treatment if it doesn't show out to be very good. So just to recall, that was decided after everybody was enrolled. That's what the data looked like. You don't think we could have decided earlier, right? It must have looked a lot like that halfway along. So why did we waste time? Isn't there a way to develop a rule that say you should quit if you aren't having an advantage? And we added that too. All right. So what we're going to do is we have interim monitoring. We stop when the criteria for considering a new therapy is good. Whenever we see that, we stop and we declare goodness. If at any time there shows little chance of declaring the new therapy is better, we're going to stop then too. We're scheduled to look at the data 3 times during the trial. And we have a mechanism for regularly examining the data, and we downweight the historical controls if they don't look like the concurrent controls. So there's a lot of things going on here. We did not assume that the new controls look like the old control therapy. So we checked every once in a while. We had a plan to check every once in a while. And in the worst case, we had a plan that said, listen, can't use the historical data at all. We're just going to stop using that data and just randomize patients. So this design, it has an advantage because it takes some old data in. And if it ever appears like you haven't done the right thing, you just stop and you go back to that old design. Kind of clunky, but at least you're never worse off than if you had not used the historical data at all. So it's pretty good. So this is what we did. We assigned, we were supposed to randomize to standard therapy or standard therapy. There's a star there. That's important to remember. Standard therapy star plus a multi-tyrosine kinase inhibitor. So we had a standard therapy. We had to modify it a bit. And we added historical data. How did we do this? I started to write out the equations that I had to solve. It was 2 slides long. It had many Greek letters to it, but I decided probably not a great idea because I have trouble understanding it, and I do this for a living. So what I'm going to tell you is we had a model that had 11 parameters that described how fast people fail. We had so we had 10 for each of the randomized patients and 10 for the 5 for the randomized patients, 5 for the historical patients, and 1 that said, how does history differ from current? And then we had 3 more parameters to quantify the difference, that how much people with widely metastatic disease, how much faster they failed in the new data and the old data, and 1 that talked about how commensurate they were. And then 1 parameter that measures the effect of the new intervention, right? There's only 1 parameter because there's only one comparison to be made. All right, now to analyze one experiment, 1 set of data to do this, all this fitting, took 2 minutes on the high performance computing facility at USC. So this is very computationally intensive. Billions of calculations to take the data set and turn that picture into that one we saw that had the hump, the line that said, this is a good therapy, this is a bad therapy. So how did we figure out, remember, because what I haven't told you is, what did I set for a good therapy? What did I set for abandon this therapy? How, and what's the randomization fraction? I had to figure that out to get a good design. And what I did was I created a few thousand synthetic experiments that took into account what the new therapy was like and what the old therapy was, if the new therapy was effective, if the new therapy wasn't effective, and what happens if history is exactly like controls, if history is worse than controls, or if history is better than controls. So I had to examine all of this and make sure the design was good. All right. And this is what we ended up with. And I'll tell you how much it cost in terms of time and personnel time. We saw that this Bayesian design, if we had designed it with a standard design, we would have had to randomize 360 patients, and it'd be 6 years before we knew the final answer for 1 intervention. By incorporating history, instead of having 360 patients, we were able to get 218 patients, a 40% savings, and faster, right? We are done in 4 years rather than 6. So it's a good design, and it even takes into account what's happening with historical data. So we thought this was a pretty good deal. And we can tell if there's drift, if the new controls are more risky this is if the new controls are the same as the old controls, we have power, the same power we would normally have. We get a little decrement in power. And if the new therapy doesn't work at all, we almost never promote it to be the new accepted standard. So this design has some great properties to it. Now, you'll notice that when we went into this trial, we had 298 historical controls, and they had over 160 events in that control sample. So what you're seeing is the controls, they don't count, like they're less value, because instead of only using half the patients here, which I probably would have been the way if the controls were equally valuable to this history was equally valuable to the new, is you have to discount. You have to be ready to say those historical controls might not be worth as much. And there is a mathematical way to do it. All right. How much did it cost to do the design? Right. So there's some big costs, right? To do just the investigation of how the design should be, it took 350 hours of computing time on the high performance computing system. The standard design that I did took 30 minutes. 20 of it was typing the data in right. 5 minutes was going to turn the coffee pot off that I boiled over, and the last 5 minutes was writing it up. So it takes much less time to figure out what to do. The programming and interpretation cost about 120 person hours of work to take the data and turn it into a real design. But if you look at it, the research costs for getting the design ready are less than one patient cost to cover for the, so it's a good deal, right? Here's probably the worst thing you'll ever have to do with. You have to deal with a statistician as a co-investigator from the get-go, probably enough to make people pale right away. But we kind of like it, so you should incorporate knowledge with us right away. All right, what happened? So we were all ready to go, and the design didn't go forward. Why didn't the design go forward? Because somebody on the pediatric and adult solid tumor steering committee looked at the modifications we had to make with the standard therapy and said, you are not treating patients well because you're reducing a therapy that works already. So we can't let you randomize to that therapy you wanted to use. So we had to totally change the design. What eventually we did was we treated all patients with the standard therapy through the end of local control, and then we ran to 2 different therapies. And that study, instead of taking 5 years, is now going to take 7.5 years to get done. So I wanted to be victorious, but I'll have to wait. It's not going to happen. All right, but there have been people who showed that this design works. Now it's all like backward looking. It's all retrospective. These authors showed that they took some published data from two trials, the Azithro trial and the CLL7SA trial. This is a supportive care trial, and this is a trial that showed Rituxan was a good thing for non-Hodgkin lymphoma. And these authors showed that if they'd obtained data from concurrent controls, first off, this Azithro trial, which actually the new therapy turned out to be more dangerous than the old therapy, they would have stopped 6 months earlier, and they would have stopped and promoted Rituxan to an effective therapy about 1.5 years earlier, had they had this historical data and borrowed it. So there's a good reason to want to do this and to do that work to make it happen. All right. Some methodological things. Remember I talked about these guys, they use some pretty loose criteria to say what's an effective therapy. We used a very stringent criteria. In order to accept the new therapy, there had to be a 95% chance that the therapy worked. We abandoned it if the chance that the therapy worked was only 25%. We decided this over a lot of simulations, and part of my work as a statistician is to help figure out what those right values are. So I still have to do that. That's my job. Well, my job is my graduate student's job because I don't do that anymore. So they have work to do to get their dissertations. Now, the reason I bring this up is we all like 0.05, right? Has to be 0.05 or it's not a study. Why? Why is that? right? Other than there's some dumb guidance at NIH that said we won't license the drug unless P is less than 0.05. Now, we know that that is actually the wrong thing to do, right? There are investigators in the literature, Spasto and Stram and Marie-Cécile Letele and Dan Sargent, they've shown that in order to make progress over a series of trials, not one, if you talk about how can you make progress if you're going to examine again and again and you want to make progress over 25 years, don't choose 0.05. It's wrong. Choose 0.15. You get good protection from accepting bad regimens, and you make progress. You make things happen. So maybe your own therapy, maybe your own study looks a little weak, but part of it is we as investigators have to be happy when somebody says, I got a great result, P was 0.12. You cannot look at them like they're crazy. It is the right thing to do. We just have to get our colleagues to want to do that. So I have some work to do to see, is this actually the right one? Did I choose something that was too stringent? I'll figure that out. All right, what do you have to do, right? You have to give me historical data. That's what we're missing. So you guys out there who have data sets, who have trials, you need to put them in a place where I can find them. So that's your job now, so that I can do my job. So Sam Bolchenbaum, I think you know Sam Bolchenbaum, because I think he talked here. He is the head of the Data for the Common Good, and he provides a portal where people can deposit data and that you can request it. So some things here, I'm going to talk about some sources. Data for the Common Good, it's a repository for clinical trials and registry data. So as we call it, real world data. There's a way to handle real world data like this trials data. It's not quite the same, but it's okay. There are standardized dictionaries in Data for the Common Good. It's anonymized patient data. Now there's an intake process, which means this is not free for the depositor. You have to work to make it happen, right? And it's always an issue, right? I'm going to do all this work. I'm going to put it in a repository. Maybe it will work out. And that's the risk we all have to take. If we're going to make progress, we all have to put something on the line, and it's our time. And these are patient level data. And you can get the patient level data, but you have to have scientific review. Another one that I think is a good source for people who want to do these trials and develop these data is the NCORP NCTN data archive. And anytime you read a publication that's supported by the NIH, that all the patient level data to reproduce everything in the study, that patient level data is there. And it is available for free. So that's something you can use to populate these things too. But it requires a proposal. And the other kicker is the requester has to enter into a data use agreement. So you actually have an institution to do it. You can't be on your own. You have to have your own lawyers and dealing with lawyers. Another thing. All right, and the last one is many of us have our own institutional repositories. I think that's a good way to go, right? Because you guys now, you're actually putting in data that you can get. I wanted to point out an example, which I think if you can do it here is a great example to follow. It's the COG study APEC 14B1. It collects data prospectively. There's a large cohort of patients where you don't actually tell you you don't have to give the therapy, you don't have to, it's not directed, you just tell us what the therapy is. We record the data and the outcome. But the other thing is, these can be linked to stored biological specimens because, of course, one of the problems is that many studies nowadays require biological verification as a price of entrance to the trial. So these studies are good because with some money, a little bit of money, you can get the biological specimens, you can test it, and you can get historical data that work for your trial. Here's the advertisement at the end, right? I have work to do in order that we can develop these methods. So it doesn't take 2-person weeks to make the design. It's bad enough when I have to take 20 minutes to do a design. People are always bugging. But it will take time. And the other thing is for you guys to help out, right? Get me historical data. I can handle it. And I have to tell you now, historical data is not worth full value. I, as a statistician, have to allow the possibility that it might not be worth it because the historical controls differ, but I can do it. I know how. So I've done a lot of this work. So let's all get together because we have this huge data resource, and pediatric cancer is a rare indication. The only way we're going to make progress here is if we all cooperate and make this happen. That's all I have to say. Thank you.
Thank you, Mark, for that passionate talk. You made statistics quite palatable and understandable for us. I think we'll take questions at the end, if that's okay.
Yes.
For the next talk, we have an online session. It's on developing evidence-based resource-adapted guidelines by Nikhil Bhakta. Yeah.
Hello, my name is Nikhil Bhakta, and I'm a pediatric oncologist at St. Jude Children's Research Hospital. It is my great pleasure to be presenting the ARIA Guide program to you today. I apologize that I cannot be there in person. If you're looking for somebody to blame, go ahead and blame Venkat because he actually pulled me over there last month, and it was just a little bit too much to get back over there with a few weeks turnaround. But I do miss being with all of you today, but I also know that you have many in the audience who can answer many questions about ARIA as well, just as well as I can. But to really, my goal here is to really introduce the program and to highlight how this tool can also be used, especially through the other talks, around implementation and research and as well as just improving care within many of your contexts. So taking a step back, what is ARIA and why do we have it? So ARIA is a program focused on the development of guidelines. Why do we need global guidelines? And I think it's always important to think about definitions a little bit. So when I think about what are the levels of guidance that many use to treat children with cancer, and India is a unique place because you have INFOG, and I think over the last decade, the effort put to actually develop these committees within INFOG around diseases has made huge strides in that country. But globally, many do not have access to protocols like you have in INFOG. And I use the word protocol because those are research or quality improvements that efforts. They're really designed to ask something or test something. And so as a result, many will say they use a protocol, but in reality, what they only have access to is a piece of paper, like a letter or a publication for how to manage a patient. So they have a table that may say, here's the drugs that you use. But we know that management for childhood cancers is truly a complex health intervention. It's not like management of hypertension or hyperlipidemia, which may be 3 to 4 pages as a guideline. Usually our protocols are hundreds of pages, and they have everything from the diagnosis and risk stratification, staging, to the treatment and management, the dose modifications, all the way to survivorship. And of course, that doesn't include the surgical recommendations and the radiation oncology recommendations, et cetera. So all of that has to come together into a comprehensive data set. And so a lot of times, most of the world is using these kind of 1 or 2 page treatment plans, but everyone aspires or says that they're using a protocol. And that's oftentimes leads to misunderstandings. And what we lack at a global level is that we don't have global guidelines. We don't have a standard repository of where you can go and say, this is how I want to treat someone. And yes, SIOP had the old ATRs, but those were really publications. They didn't really tell you, as like a protocol does, how to manage a patient comprehensively from A to Z. And so that's really the space that ARIA is trying to fit. How can we give you the best evidence that's contextually appropriate to comprehensively manage children with cancer for all diseases, supportive care areas, palliative care areas, and survivorship? And so with the Global Initiative for Childhood Cancer, within that cure-all package that I'm sure everybody is familiar with, the R stands for regimens, which is really a proxy for guidelines. And what we've seen is an outpouring of interest by countries and hospitals to develop their own guidelines. And if anybody who's worked on a guideline, you know that that's like it's usually averages about 3 years to develop a guideline. And because you have to do the evidence synthesis, the evidence decision frameworks, you have to make sure you have kind of a voting process, make sure it's contextually appropriate, have patients with lived experience involved, all kinds of processes that people often skip. But to really call it a guideline, you have to do. And so if you do it properly, it makes no sense for everybody to keep doing the same thing over and over again, especially when we realize that at a global level, we can develop this and realize that there's more in common between us than there is different, so that we can actually develop guidelines together with multiple different perspectives and then work to adopt and adapt them at the regional, country, and hospital level. So rather than every country develop their own guidelines, let's work as a team at a global level, but have representation that's global in that development process. And then spend more of our time actually studying how to implement those guidelines rather than how to actually develop them. And this is very important because it reduces all of that inefficiency and allows us to take and harness opportunities to innovate and allows for people to study how to use guidelines more appropriately in their context. So I'm going to give you a little bit about like the ARIA guidelines, what is the collaboration, how are the guidelines created, and then a little bit about how we're disseminating them. So ARIA itself is a collaboration between St. Jude Global, SIOP, IPSO, PROS, and CCI. And it's a collaboration between each of our organizations, recognizing that no single organization brings all of the tools that we have that are necessary by themselves. And so we started this by forming a steering committee. And really, I want to thank Raman for being one of our initial founding members of our steering committee. And then, of course, now Rashmi has joined the steering committee after Raman left. And so, India has incredibly contributed to ARIA, both in terms of that leadership level, but then also at the development level. Underneath the steering committee, who really advise, we have the directors, myself and Michael Sullivan, who from the SIOP side. We have liaisons from each of the professional societies who make sure that we incorporate their perspectives in everything that we do. And we've developed what's called the coordinating center at St. Jude, which really helps to keep all of the wheels moving and all of the efforts and processes happening. These groups at a programmatic level feed into these global panels that we create. So there's working groups, such as small groups that really help us to develop the strategy of how to develop the guideline, and then global panels of 70, 80 people from all regions, multidisciplinary, and I'll get more into that in a minute, that really help us to think about what is the guideline appropriate. And to draw those individuals, we have over 80 collaborative groups around the world that collaborate with us to nominate individuals. And then once a guideline is developed, we also pull in external experts to make sure that we don't over-adapt. And so all of this is a governance structure that we've developed and put in place for each guideline ensures that it's at the highest quality. We also have the WHO that, as a non-voting member, is asked to participate in our steering committee meetings and also align our efforts with the cure-all efforts. And so if you think about what is the process that we use, the first thing that we do is we took a first step at understanding what are the content needs. So what are the disease-based guidelines we need and what are the non-disease-based guidelines we need? And this has been a lot of work because, for example, we often say supportive care as pediatric oncologists, but I would challenge each of you, what do you define supportive care as? What are the topics? And so we actually did this with 80 pediatric hemoncs and other healthcare providers from around the world, and to find out what does supportive care mean topic by topic. So the first was defining the content needs that we needed. And then thinking about how can we develop that content. So we've got, like I said, the central coordination from the ARIA Coordinating Center, and then community engagement across the world. And we've had over 600 healthcare providers already involved in ARIA development, and with representation from all 6 WHO regions and numerous countries from around the world. And when we think about who are participating, we kind of have these bins. And so we have content experts. These are, again, these clinical trial groups, many of the INFOG members who are really experts in a disease field. But then we also have context experts. They may not know everything about rhabdomyosarcoma or Wilms tumor, but they certainly treat a lot of it in their context. And so they can tell you what's practical and what's not practical, and also raise questions about whether something is really appropriate or not for their setting. And so we see expertise and value in both of those types of individuals and incorporate them into our review panels. Also, of course, making sure that we have multidisciplinary review perspective, survivors and families with lived experience feed into each guideline, and then young investigators we try to bring into the process. And so this is just a small list, but it tells you like who are the types of individuals or organizations that we tap into to give us nominations for each of the ARIA guidelines that we develop. And so then what happens when we develop a guideline? it really starts with kind of an evidence synthesis process. So what is defining the standard of care at any context? So what is the publications that exist that are out there? And then and then understand how have people adapted based on that. So it may not be that they run a randomized controlled trial for how to manage Wilms tumor without radiation, but there are publications out there that have talked about that either against historical controls or a case series. And so we try to look at what adaptations folks have done and pull that evidence in as well. And then when evidence is poor, we utilize consensus surveys or Delphi methods in order to be able to incorporate that into the adaptation process. And so we have a robust methodology that really sequentially goes from the highest level evidence to then understanding where there are gaps and then incorporating the panel that I just described, making sure that they are involved in helping us to come to consensus on what is, at least to the best of our knowledge, the safest and most effective way to treat. And of course, all of this is done underneath that broader perspective, as I highlighted, that governance model and standard operating procedures that the steering committee and the ARIA coordinating center help us to do. And then from there, really, we take the guidelines, because a PDF has never saved a patient's life, and think about how can we disseminate that into decision aids as well as ready-to-use dissemination tools on the web platform. And that really is what that last part of content delivery is, where ARIA Guide. And we use a lot of user-centered design. We go to our colleagues, many of you in the audience, and ask you, how does the website make sense? Does it work? And I'll get more to that in just a second. But first, just to highlight, we have over 123 guidelines that are planned, 54 are in progress, and 22 are actually already on the portal. I think 23 now, actually, and are constantly being developed. And so on the platform itself, what do I mean by that dissemination portal? And so really, ARIA itself is a novel tool. There isn't anything quite like it in the global health space. And so we're really excited that we've been building this out, and we're excited for many of you also to be able to play with it. So you can go on there even today. It's freely available at ariaguide.org. And if you do, you'll see a landing page. And so, what will you find on there? You'll find guidelines with a bunch of with narrative details, resources and tools, regimens, checklists. It's freely available, as I said. It's aimed at pediatric hematologists, oncologists, pediatricians, trainees, but also allied healthcare workers, so nurses, social workers, pharmacists. This is meant to be anybody in the healthcare field that would benefit from being able to see where patients' treatment may lie. There's no protected health information in this, so you don't have to worry about like IRB or ethics review to be able to access this. And but we do ask for anybody who uses it for their consent, so that we can track their user metadata to be able to understand how the platform is being used, so we can improve it as we move forward. So, I'm going to give you an example of how this platform, how it can be used. So, Hodgkin lymphoma, we know is there's many ways to treat it, especially what do you do if you have CT and PET scan like we do in the US? Or what happens if you don't have PET scan and only CT? But then what about when you don't have radiation and you don't have a CT scanner? So you can think about each of those are different treatment strategies that we have to have. So one guideline will have multiple nested guidelines in it for how to manage a child in those contexts. And of course, we know there's various forms of radiation, 2D, IMRT, proton, et cetera. So how do we link all of that? And you can see it can get to be a total mess if you were to use a PDF to try to manage that patient, which many of us do for the protocols that we use today. And so this is where the ARIA portal can be useful. If you click on a selected disease, you'll see that there's Hodgkin lymphoma there. You can click on that. You can also go to the library to get the full PDF version of that as well. And if you click on Hodgkin lymphoma, for example, you can click on pre-evaluations. It'll tell you what recommended clinical testing you need, clinical evaluations you need to do, what labs, what imaging that you might need as well. There are risk staging system classifiers in there, so that within a few clicks, you'll be able to get the stage. So if you forget your Ann Arbor staging system, this is especially useful for trainees. They can click. If they click on the yellow pieces, it'll actually give them information. So that way, they don't have to be an expert in understanding what bulky tumor means. They'll give them information on what that looks like. And then if you click on the treatment, it'll actually give you these roadmaps. And so you can click between strata. The system should automatically know for what resources you have at your hospital and tell you what the strata is that is best. But let's say you actually would prefer to do a different one. You can actually change that on the fly, and it'll give you different roadmaps, as you see here, that if you drill down into those, you could actually then get access to the principal chemotherapy and alternative chemotherapy regimens. You can enter in the height and the weight, and also dose modifications, and it would automatically calculate all of that for you in real time so that you'd be able to quickly make decisions. We have the same thing for things like supportive care, like PJP prophylaxis and other wells as well. And so with that, I want to take a chance to again thank the many contributors who are part of our coordinating center. I want to thank again many of you from India, from INFOG and from India in general, because we have tapped into many of you to support us as panel members, as working group members. Deepak, Amita, and many others. And I won't be able to, I apologize if I didn't call your name out, but there's many of you who have been really advocates and leaders within our ARIA ecosystem. And so thank you to all of you. And again, apologies for not being there, but I look forward to the question and answer session to follow. Thank you.
Thanks, Nikhil, for taking us through the ARIA guide framework. We'll take the questions at the end of the session. Moving on, we have the third talk for the morning session, Assessing Cost Effectiveness of Novel Therapeutics by Dr. Manju Sengar, Professor and Head of Medical Oncology, Tata Memorial Hospital.
Don't want to look very tall. Very good morning, everyone. This is going to be a little different talk than what all of you have heard, and I think it's rightly been placed on Sunday morning. Hopefully, all of you have slept well last night, and I don't put you to sleep early morning. So what I'm going to cover today is a different topic, which is important, given not so much in pediatric oncology till some time back, but more in adult oncology, where novel therapies get added day in and day out. And it's a limited kitty, which is available to fund many of these treatments. And it's very important in our setting now, as we are moving more towards including many of these drugs in the universal health coverage package through the Ayushman Bharat scheme. So what is, health technology assessment? It's a systematic, transparent, unbiased process which decides whether an intervention is giving you value for money. And it just not involves the data from clinical trials. It considers cost, it considers the societal impact, the economic impact, and all that is put together through a systematic process to decide whatever is the research showing, whether it's worth bringing into the policy or not. So it's linking the intervention which is derived from research, a good clinical research, translating into a policy and seeing whether this particular health intervention, it could be drug, it could be a surgery, it could be radiation, is giving you for value for money or not. There are a lot of things which are taken into account when we talk about health technology assessment, because the ultimate idea is that you want to expand your universal health coverage and bring in an intervention which is giving you, whether it's optimizing the use of resources or not, whether it's, worth bringing that enough data in terms of clinical impact. You want to ensure that there is equity in healthcare, and all your decisions are evidence-based. So it takes into account the data. And of course, in any setting across the world, cost control becomes an important part, because we can't fund everything which is available. And it definitely takes into account, that the technologies which are being recommended are safe and effective, and improving the quality of care as we go along. So typical things which are taken into account when the HTAs are done, comparing two intervention, is you first and foremost is whether there is efficacy and effectiveness or not. What's going to be the value for money? What's the budget impact? what's the societal, legal, and ethical consideration of recommending or not recommending an intervention? And oftentimes these two interventions, or sometimes more than two interventions, are compared to decide whether you want to recommend a technology as cost effective and thus to be included in the universal health coverage. I'll give you a bit of background, why all this, what we are doing. So some of you are aware that National Cancer Grid is a network of cancer centers. It was established in 2012, now expanded to include almost 360 plus cancer centers, research institutions, and organizations. The mandate with which NCG was started was to ensure that anybody in any part of the country, irrespective of what their social or economic status or geographical location is, they get uniform cancer care. A lot of efforts have been made in terms of training individuals, in terms of doing research which is relevant to our country. But one thing which has been there from the beginning, as you heard in the previous talk, is the guideline. What NCG started was a resource stratified guideline. And there were three sets of guidelines because we know that there cannot be a single set of guidelines applicable across country. So there were essential set of guidelines, that is the minimum care which needs to be delivered. Then on the other spectrum, there was optional set of guidelines, which means the best evidence which is available and the patient should be able to get that irrespective of cost consideration. The middle set, which was the optimal set of guideline, included the best available evidence and the cost effectiveness. And that's where we realize that though we have put this in resource stratified guidelines, a lot of it is just a subjective cost effectiveness, a little more improvised version of GOBSAT, where people sat together and decided that whether an intervention is cost effective or not. So we needed processes to bring in the guidelines to ensure that what we are recommending as optimal is cost effective. And therefore, we needed the health technology assessment. But the problem of health technology assessment, you need people who can do economic evaluation, who can sit down. You need methodologists who can collate evidence. Then you need people to appraise that evidence. And then once you have that, you should have a process how this HTA is going to get incorporated into day-to-day practice or even in your recommendation. And the major challenge with us was not having this expertise, as well as within the country not having good quality cost data or the costing systems which are existing. There's so much variability. And the applicability or the generalizability of data which is there from clinical trials into our setting. So that we can't use the same data because our population is different, the intervention or the comparators which you normally use are different. And in any HTA, it takes roughly around a year, if not more, to do this if you have a dedicated team within the group. And we realized that we had close to 150 interventions to be evaluated. So I think it's our next generation, and even may not be the next generation, who can bring in this HTA evidence into the guidelines. So we resorted to something which is different, which is becoming more and more common thing, even in these settings where there is a full HTA capability available. That's adaptive health technology. What is the difference between an adaptive HTA and an HTA? Adaptive HTA is something which utilizes the available international evidence, both for the clinical and the cost effectiveness, and not just one country, multiple HTA agencies if they have done it. It helps to bring in the cost effectiveness data from the other country, and I'll show you the process, what we used into our own context. And therefore, what it requires is lesser number of hours, lesser number of people who need to be trained, or it's a faster training. A week before, we have imparted this training to close to 25 people within the country. These are clinicians, so it's relatively easy to train for. And therefore, you can make decisions little more urgently as we need it for our guidelines. However, it's not that everywhere we can utilize adaptive HTA. Three things are very important when we are thinking of moving away from a full HTA to an adaptive HTA, is that the question has to be simple, meaning it has to be clearly defined question. Your population should be clear, intervention should be clear, comparator and outcome. There shouldn't be no clarity in that clinical question. There should be certainty of evidence in terms of good health technology assessment reports available from certain international agencies from any country, and of course the urgency. And that's what decides whether you want an adaptive HTA or a full HTA. So we followed typical health technology assessment process, the full HTA process, which requires a topic selection, topic prioritization, then, producing a scope, then doing evidence review, recommendations, and then implementation. So I'll just take you through, some of these steps. So as the slides are coming up, I'll just, continue blabbering. So, so if you look at topic prioritization, that basically involves how common is a disease in your country for which you're trying to, bring in this intervention, how good the data for intervention is, what is the estimated cost of this intervention, how this will translate into budget impact for other technologies which this particular technology is going to replace. So just to give you an idea that if you're bringing in an immunotherapy in our guidelines, how many maternal and child health intervention this technology is going to displace from that same limited pool of money. So what we have, what we call as opportunity cost, which is going to come in. And yeah. And whether you have the capacity within the system to deliver this new intervention, which is be it surgery or proton therapy, or for that matter any new drug, whether your system has capability, if you bring it into guideline, whether everybody will be able to apply this technology even in the remotest part of the country, or you would need special investment in the infrastructure to develop to ensure that this technology is implemented. And who's requesting it, whether it's coming from policymaker, pharmaceutical company, clinicians, who's requesting, and based on this, a topic prioritization list is created. We then produced a scope. Scope is nothing what we typically look in our clinical trial when we create a population, intervention, comparator, and outcome. But here it becomes very, very important for two reasons. One, you're going to search data using this PICO question. All your subsequent review literature is going to be based on this PICO. And if your PICO, what you have created, there is no study or the international evidence is not done using this PICO, then you can't do an adaptive HTA. So for these two reasons, it's important to make sure that the PICO is written correctly and it's vetted by the clinicians who are involved in developing these clinical, adaptive health technology assessment policy briefs. And then you bring in that question. Now, there may be trials who have done using this PICO, but that's not the question which you are asking. Your comparator is completely different. Again, you can't use the evidence from the literature or from the HTA reports to do this adaptive HTA. Once we have the PICO, which is created, a rapid literature review is done, a price benchmarking analysis, and the treatment cost estimate. So when we do this literature review, three things what we want. We look at all the international HTA reports which are available. We extract the data in terms of clinical benefits, safety, outcome, survival data, or quality of life, and then contextualize whether the same clinical trial is going to be applicable to us or not. People included in that clinical trial, are they the same people what we see in our country? And then the price benchmarking analysis is done. Nothing, we just, try and see that the price at which this particular drug is being made available in India, we correct it for the GDP purchasing power parity and use this correction factor and come out with a cost and say if this was the cost in our benchmark country, whether that country would be including the drug into the universal health coverage or not. So that's basically the price benchmarking analysis. And once we have the price decided, we look at that if you were to include this drug in the universal health coverage, what will be the budget impact? And for this, we take into account what's the prevalence, how many number of patients, how many number of cycles which need to be given, what additional resources would be needed, and what would be the cost of that. And then for us, the benchmark was relatively easy. We know that for PMJ, it has to be under 5 lakhs because that's what the annual coverage is for the family of 4. And if it was higher than that, then in that case, we said that the budget impact is too high and that cannot be included in in the guidelines. And then a recommendation is made with respect to the cost effectiveness. And this is basically this entire data sheet, which contains all the information what I have listed earlier, is presented to the clinicians. Then a polling is done, which is basically an anonymous polling so that people can say what they want. And this just not looks at cost, clinical effectiveness, but a lot of things, whether this will be applicable to their own setting where they're practicing. And all those discussions are then made once the polling questions are available. And then the decision is made in terms of whether it's cost effective, not cost effective, meaning the costs are outweighing the benefits, or there are other options which have lower cost, and whether there will be a discount needed for it to become a cost effective, or it's unknown that we don't have enough data to make recommendation, or you need a full health technology assessment for this technology, and therefore we have screened this out. And all this is presented in terms of policy briefs, which we put out on our website and definitely sent to now, because we have become a regional resource center for health technology assessment India. We send out these policy briefs to them, including all these points in terms of literature review, the price analysis, cost, and other consideration, and what was the final conclusion. So we did 10 technology assessment as part of this initial exercise. This started in COVID. We needed to learn something different. So we did these 10 technologies, and this included checkpoint inhibitors like pembrolizumab, CDK4/6 inhibitors. It included osimertinib, nimotuzumab. We did try to do a non-pharmacological intervention assessment. So we tried to look at robotic prostatectomy, and we tried to look at hypofractionated radiation in breast cancer. And there were challenges for the non-pharmaceutical interventions. We were quite, clear in terms of the pharmacological interventions. And just to give you an example, it only told us that you will need, the cost of pembrolizumab in India, if you correct it for the international benchmark countries, was almost 15 times higher. Though we consider that, Indian costs are cheaper, but when we do the analysis, we realize that this was so much higher. And if we were to include that, the annual budget impact is going to be way, way higher, and it's going to replace practically everything which is there in oncology if you were to pay only for pembrolizumab. And that's how, once the assessment is completed, the idea is that it is included in the guidelines. And since we have a memorandum of understanding with the Ayushman Bharat Pradhan Mantri Jan Arogya Yojana, that is run by NHA, whatever is listed here, the idea is that it goes into the health benefit package, and therefore it gets implemented. So there is some institutionalization of how we are going to use this. And as I said, that it's not very easy. It looks, that it's the best way to do. Why one should be wasting time in doing full health technology assessment, at least in countries like ours. But not always that's possible. You may not have the data for your intervention, what you're looking for. It may not be relevant for your country, or the costing which is needed. And that's largely true for non-pharmacological interventions, because for drugs, the majority of the cost is for the drug. And for the rest, it becomes a little easier. But when we are looking at radiation, we are looking at surgery, there are too many factors: expertise, consumables, operation theater time, delivery time. There's so many factors which are involved. And therefore, doing this for non-pharmacological intervention becomes challenging. And that was our experience too, that for this, we would need to move on to full HTA or have costing systems developed in-house within National Cancer Grid to ensure that what we recommend as cost-effective or not cost-effective has meaning. And that was my last slide. Thank you.
Thank you, Dr. Thank you, Dr. Manju, for such a relevant talk. We'll move on to the next talk, sticking to time. So may I invite Caitlin Duffy to talk on translating evidence into practice, implementing novel therapeutics in LMICs. So Caitlin is an instructor at St. Jude faculty, Global Pediatric Medicine. So welcome.
All right, thank you very much. While we're waiting for slides, I just want to thank the committee for inviting me. This has been such a fun conference, and thinking back, I don't know that there's been a single day where I've just gotten to sit and think about immunotherapy for a full day and then follow that with practical applications. So thank you very much for having me. So today I'm going to talk about how we are translating novel therapies into varied resource settings. And from a disclosure, so I did receive an unrestricted grant from the Amgen Foundation previously. I do lead the St. Jude Global Program in the BlindCyto Humanitarian Access Program, and with that, provide in-kind support for their medical expert panel. First, I'm going to talk to you a little bit about the process that we've taken to actually translate these novel therapies into practice. And then the next step, which is really how do we then take what we've put into practice and create this new practice-based evidence, and how can we use that? So to start with, blinatumomab, which we have talked a lot about with this conference, is an evidence-based intervention. And I think this is a really cool figure, but what I'm trying to show here is that like this has been a very long process in terms of generating the evidence for this type of therapy. And so you can see it really was first presented to the scientific community back in 1997, and it actually took 17 years for it to first get FDA approved. And then about 4 additional years where there was data building from randomized clinical trials that then supported extension to a broader salvage therapy. And then this past year has been really exciting for blinatumomab because we actually saw translation from what was historically just salvage therapy now into the front line, both for pediatrics and for adult therapy. And so really a drug that for kids, especially in the last COG study, showed an 8 to 10% increase in survival just based on this one therapy. So really with that making a big impact and quote unquote, paradigm shifting impact in our therapies, which is great, but how do you then implement these types of drugs? And I think it's pretty conservative in saying that when you go to think about how you administer and manage blina, this is a complex process. It is a very long cycle, so it's a continuous infusion medication, has to be given 24 hours a day for 28 days in a row. You need a dedicated central line. You have to start these patients inpatient. If you have a robust outpatient infrastructure, you could transition them with routine follow-up. But it can be quite challenging, especially if there's any kind of interruptions in care. And so otherwise, it can be given as a full 28-day inpatient treatment. You need a lot of coordination from your multidisciplinary teams. You need very unique medical supplies, which often are not available unless you are actually giving this type of therapy. There are very unique nursing procedures that if you don't follow can actually risk precipitating an adverse event, so very serious. And you need consistent long-term follow-up because some of the adverse events and some of the long-term side effects for these patients can increase their risk for infections. And these side effects that we talk about are rare, but it can occur very seriously. And actually by intervening, you can reduce the severity. And so there's a lot of complexity with how you manage these drugs. And one of the unfortunate realities is that despite its efficacy, there's very limited global adoption. So this is not even a drug that we give at all of our affiliate centers at St. Jude. And there's very limited accessibility and commercial availability worldwide. And that's really unfortunate because what we're seeing now is like we talked about this process of discovery and integration into quote unquote this standard of care, but failure to translate it broadly puts us in this place where we have this discovery delivery delay, which really can contribute to a survival gap. Which is unfortunate, especially in varied resource or low and middle income countries, because there's actually what we call kind of this relative advantage. And so it's this idea that we know the populations that we're often dealing with, there are reports that show higher rate of high-risk genetic ALL features in a lot of these populations. We know that with some of the toxic blocks for relapse therapy, there's a lot of toxicity that's associated with it. And while we talked about some of the unfortunate side effects with blinatumomab, we actually see less of fever neutropenia, less sepsis, less mucositis, less infections compared with traditional chemotherapy. And often in some of these settings where we're using adaptive treatment regimens, there might be a higher rate of relapse and unfortunately very limited options for cure after relapse. Because transplant requires a lot of infrastructure, is exceptionally costly, is a significant treatment burden to the families. And immunotherapies often are not registered in a lot of countries. Even if they participate in trials, we know that those drugs aren't readily available afterwards. And so, we talk about it's great that this drug is efficacious, but it's very complex to give. It's very costly to give. And so how do we think about doing it really intentionally? We did try to think like, what are the examples of how this has been done in the past? And I think we all kind of know this historical context of this being done in not the most appropriate way. Lots of issues related to power differentials, lots of ethical considerations. And this is a publication from a drug donation program that donated imatinib. And they said, even though we had success with this program, there were a lot of common challenges that we encountered: low priority across many governments, very limited health infrastructure that supported how we identified these patients, cultural and educational differences, and issues related to financing. And that's just for like an oral medication, not one like blinatumomab blinatumomab, which we've talked about, has so many other things that we have to consider. And so we really recognized that we needed a different approach if we were thinking about how we were going to successfully translate this type of therapy. And so we needed this kind of multi-level, multi-phase solution. And so to first address some of the issues related to cost, the regulatory components, the logistics, we created a cross-sector collaboration called the Blincyto Humanitarian Access Program. This is a drug-only donation program that is a collaboration between Amgen, the pharmaceutical manufacturer, St. Jude Global, and an NGO called Direct Relief, who is an expert in drug importation. And so the mission of this drug donation program is to create free access for institutions in low and middle income countries where it's not commercially available. And I have this little figure to just say that this was a very big effort of like trying to figure out how we align all of these different missions, because we know that everybody has kind of different objectives, but really trying to bring people together so we can highlight and act in that center where there is alignment and where we can do good. And we recognize there were other types of challenges, like we talked about, related to administration. And so to think about how we can further support institutions, the staff working there, think through how the complexity impacts how it's delivered, we created a hybrid effectiveness implementation study. And this is an implementation science-based approach, which is all about answering the questions, how do we get what works to people who need it, so that we can minimize that discovery delivery delay? And how do we generate this really important real-world evidence, which Manju was just talking to us about? And then to highlight, you did not have to participate in the research study in order to participate in the drug donation program. 6 of our 13 institutions in the drug donation program ultimately participated in the research study. This is a conceptual framework that kind of talks through the different phases that we took. And the first box really just shows you that we spent a lot of time trying to understand where are we going to be implementing this drug and what about it do we need to be considerate of? And then what strategies are we going to use to support implementation? And then we went through a very intentional evaluation period with the ultimate goal that what we come out of this at the end is not just something that helps us implement blinatumomab, but also could potentially be used to implement these other novel emerging therapies. And this work was supported by the Conquer Cancer Foundation, ASCO, and the Amgen Foundation. This is a little bit more detailed figure that just kind of shows like those implementation strategies, those like, how did you practically go through the steps of creating this process? And so you can see what we did to engage partners, what we did to collect baseline data and plan our strategies, and then how we monitored implementation and thought about sustainment. The first thing we did for an implementation strategy was to develop this multidisciplinary training toolkit. We did a comprehensive evaluation of what do you need to be considerate of and how you administer Blina. We looked in the literature, we looked at our own institutional practices. We also did a hospital readiness assessment questionnaire, which helped us understand what are going to be common challenges we may run into with some of the different resources available at centers that we're partnering with. And then we did a planned adaptation and created guidelines that would help us understand how we could support physicians, nurses, and pharmacists and make sure that all of those materials speak to each other, because I think we've all experienced where one person has something that gives you instructions, somebody else has something that gives them instructions, and they're not the same. And also created a resource so that clinicians or nurses could really walk through the process with patients and families so that everybody was informed. Here's some examples of some of the materials that we've created for physicians, for nurses, really just trying to recognize, like, not only give you a lecture, but also think about, like, what do you need in your hand at the bedside so that you feel comfortable as a provider taking care of these complex patients. And we created kind of a 3-pronged training approach. We did this at the beginning of COVID, and so we pretty rapidly converted to a virtual platform where we did multidisciplinary team training. We created a resource repository with all these workflow tools, recordings, and didactics. And then we created kind of multi-level team support. And so within each team, we asked for them to pick some like key individuals across their nursing, physician, and pharmacy staff that were going to be points of contact, and then created a medical expert panel with those of us at St. Jude and Amgen to talk through issues related to adverse events and patient eligibility. And so this is kind of the process of how we took this evidence that we talked about and put it into practice. And now really the next step is thinking about how do we take that new practice and synthesize that so that we can understand something to create new evidence And so understanding those implementation barriers, understanding how we were implementing it, knowing that you have to implement something well in order for it to ultimately demonstrate local clinical effectiveness. And that is really important because once we have that new evidence, then it helps us think about what are the next steps in order to identify strategies so that we can better support broader and scalable implementation and practice. And so this is just kind of an overview of one of the things that we did to understand implementation barriers. We used an implementation framework called the CFIR, which basically just said, thinking about all of these multiple levels, what are some of the challenges? And so understanding some of the trade-offs that we made in terms of creating guidelines for practice. Like, yes, it was safer to give things inpatient, but we know bed capacity is a significant challenge. We also used implementation tools like the implementation climate scale and organizational readiness for change so that we could understand some of those institutional factors that impact how things are implemented. And one of the things that I want to highlight is this kind of like motivation to implement, because readiness means like, are you willing and are you able? And I think one of the things that's really impactful about this project is that our teams are incredibly willing in a way that often, we're not even seeing that type of motivation with teams that we're working with domestically. So I just want to highlight like that is something that is not to be diminished and something really powerful that I think can come out of this work. This figure didn't translate perfectly, but we also looked at implementation outcomes and we used the RE-AIM framework to do that. And so we measured things like reach with all of the patients who would potentially be eligible based on how many relapses you have, how many MRD positive patients you have, how many are actually getting enrolled in this program, why are there gaps if those numbers don't match? How effective is our training toolkit? How effective is our donation program? Are the drugs getting there when we think that they're supposed to be getting there? How does adoption vary between institutions? Because we gave guidelines, but we also said, you're doing your own institutional practices. We want this to be integrated into what you do and how you naturally take care of patients. And so we want to understand how people are doing that differently and really figure out if there are commonalities between those different institutions. And then look at things that are those implementation outcomes. And so those are things like, is it feasible? Meaning, was it easy? Is it acceptable? Did you feel like this was a reasonable way to do things related to the drug donation program? And was it appropriate? Did it fit in your workflow? Did it fit in how you take care of patients? Did it disrupt, for example, like all of your scheduled chemos? Because that would be something that doesn't work. And so we surveyed multidisciplinary providers to really get input on this and use some standard implementation measures. And we presented some of this work at ASH 2 years ago and have a manuscript that's going to be coming out soon that gives a little bit more detail about this. And we also wanted to know, like, how is this program sustained over time? How many patients are getting it each year? How does that change? And then we really were thoughtful about how do we create clinical evidence in populations to show that it's working for patients in this setting, it's safe for patients in this setting. And so we were very intentional about minimizing what we could in terms of those clinical outcomes. So we wanted to pick things that were clinically relevant, that were going to be documented as part of routine care in order to really help reduce the burden on the local teams so that we could improve our feasibility of data collection. What can we plan and what can we consistently get data for in the future? And then also make sure that it's de-identified, that there's no PHIs, we have like no years, so that we could really improve our data sharing. And so this is just an example of some of the different elements that we've included. We have 106 patients that have been part of this study and are very excited to be reporting this shortly. So looking forward to being able to share this with everybody soon. This is getting close to my last slide. And so really, what are we doing with this information? We're thinking about going forward, how do we improve feasibility? How do we create SOPs to get patients out of the hospital? What do you need to support those patients? What kind of data do we need to collect in the future to really think about how we further refine patient selection? How do we create a model for scalability so that AIM-3 that we were talking about, and then how can that be a model for how we engage with industry going forward and how we measure outcomes going forward, and how can this be applied to alternative novel therapies like we've talked about anti-GD2 CAR T cells, and what strategies can we support sustainability? So cost effectiveness, things like the health technology assessment, I think would be really interesting, and creating and supporting this regional network of experts and using this real world evidence to then be further advocacy for national and global agencies to think about how these treatments can be more broadly available. And my last point is that, like, one of the big things that we're taking out of this is that it is really hard to give a continuous infusion therapy, period. And especially in these settings, we're worried about medical supplies that might not be available, bed capacity, the infection risk with central lines. Alternative administration routes make even more sense here. And so as we're seeing this field continue to evolve and we're seeing sub-Q versions be more readily available, being tested in adults, we really think that it's our role to advocate for this type of option to be available in these sites, both in participating in the clinical trials and having it be readily available after those. And so this paper, when it was published last year, talked about, you know, we hope that this could have tremendous global impact, and I certainly hope that it does, and I'm hoping that the work we're doing now is going to help it be able to do so. So really, I think that the value of this type of approach is to address that relative need for some of these ALL treatments, to identify what clinical questions we need to answer to make sure that it's right for the patients that we're giving it to in these real world settings, so that we can prevent this further widening of the gap. And I really believe that this kind of hybrid parallel research approach helps us then create the data to be better advocates going forward. So thank you very much.
The next talk, we have, it's an online session. We have collaborations in rare pediatric tumors and big data by Carlos Rodriguez Galindo from St. Jude's Research Hospital.
Good morning. Thank you so much for your kind invitation to attend this session and speak on the rare pediatric tumors initiatives that we are currently conducting in the United States through the Children's Oncology Group. I'm sorry I cannot be there in person with you due to problems with my travel, but I hope I can be live during the time of questions. So over the next 15-20 minutes, I would like to share with you some of the collaborations that we have started within the Rare Tumor Committee in the Children's Oncology Group. I am the past chair of that committee. The current chair is Dr. Ted Latch from Children's Hospital of Philadelphia, who has been very kind to share some of the slides that I will be presenting. So what are the rare tumors? We try always to struggle on how to define rare tumors. The National Cancer Institute in the US defines rare cancers as any occurring in less than 40,000 people per year. And then from that point of view, all pediatric cancers are rare. The expert consortium in Europe proposes a definition of less than 2 cases per million per year, or cases or cancers that are not considered in clinical trials, therefore more expanding to orphan diseases. The definitions are becoming even more challenging in the molecular area. For example, NTRK fusion cancers may occur 1 in 3 cases per million per year, or ALK-mutated neuroblastoma, a rare subset of a more common cancer with an incidence of less than 1 case per million per year. Altogether, rare pediatric cancers comprising most of these common definitions comprise about 10% of all pediatric cancers. And what we do know is that improvement in outcomes are substantially below or behind those of common pediatric cancers. Here I want to highlight how the Children's Oncology came together, because this type of collaborative groups are critical to advance in childhood cancer, and has been critical as well for advancing in rare childhood cancers. The Children's Oncology Group is a new formation, and we are currently celebrating the 25 years, but it builds on the history of multiple cooperative groups that started as early as the 1950s, 60s, and 70s. Some of them were comprehensive groups, others, as you know, were very disease-specific, like Newitz for Wilms tumor, or IRSG for rhabdomyosarcoma. Altogether today, the Children's Oncology Group comprises more than 200 sites and more than 10,000 members. So over the next few minutes, I will go over some of the rare studies that we have conducted within the Children's Oncology Group, provide some examples of how we manage rare tumors studies outside of the Rare Tumor Committee, and brainstorm a little bit on lessons learned, future directions, and then some of the current initiatives on big data. So the rare tumor disease are within the children's oncology group. As we said, any extracranial solid tumor that is not falling within any other disease committee, that is already included part of a rare tumor committee. And we have two main categories, the rare pediatric cancers and the adult type cancers that occur in children, such as colon cancer or lung cancer. On the right of the slide, you see the diseases that one way or another are considered within the rare tumor committee of the children's oncology group, going from adrenocortical carcinoma to thyroid carcinoma, from the least frequent to the most frequent. One of the main purposes as well of the children's oncology group is that even in the absence of clinical trials or guidelines, we have put an emphasis on prospectively build a database for molecular characterization. And so there is a molecular characterization initiative within the children's oncology group that we have assumed within the rare tumors committee with, as you see, some data on terms of numbers that have been enrolled over the last couple of years, including all rare cancers, up to 40 cases in some cases, or 30 cases per month. But then you see on the right the type of cancers that we are molecularly characterizing, from GI cancer to neuroendocrine tumors, for example, to thyroid cancer. We make an effort at making sure that each one of those cases can be characterized. The Children's Oncology Group has also a large epidemiology and data banking initiative that we call the APEC 14B1, that includes all childhood cancers that occur in institutions that have COG membership. And so it's an epidemiology study with prospective clinical documentation that at the same time collects the specimens. And so this is basically a mapping of how this has impacted in rare tumors. And we have large numbers, more than 800 cases, for example, of DNA specimens of different types of rare cancers. But we try to collect everything from paraffin-embedded tissue to CSF to urine samples in some of these cases, obviously, and then DNA and RNA. So what are the therapeutic trials that we have been able to put together to generate evidence, to generate the knowledge, to set the subsequent development of clinical trials and evidence-based therapies? So this is basically, again, the types of rare cancers that we tend to see in the rare tumors committee. And so all of them, except for thyroid carcinoma, in the 2 to 3 or less million cases per year. On the right side, you see the total number that we see in the US, in some cases as little as 2 cases per year in the US. The highest number in neuroendocrine tumors, about 200 cases per year, but the most common, like retinoblastoma, for example, less than 300 cases that we see in the United States. So in retinoblastoma, the most common of all these rare cancers, over the last 2 decades, we have been able to launch and almost complete 6 studies that include intraocular disease, so efforts at systemic therapy and local control for intraocular disease, treatments for patients with enucleated retinoblastoma, like ARIA or 332, and patients with metastatic disease. And most recently, 2 studies of intra-arterial chemotherapy for retinoblastoma and intravitreal melphalan for intraocular retinoblastoma. So I will just highlight 2 of those because I feel like these are the 2 that probably have helped us define through these trials what is our evidence-based recommendations. One is enucleated retinoblastoma that have participation of institutions in India, which is ARIA 332, which basically included patients with unilateral retinoblastoma, enucleated, then risk-based adjuvant chemotherapy that was based on massive choroid replacement, posterior uveal involvement with concomitant optic nerve involvement or optic nerve involvement posterior to the lamina crudosa. On the right, you see the overall event-free survival and overall survival for these groups of patients, basically showing that this risk-adapted therapy was able to optimize outcomes for all patients with survival rates above 97% for the entire group, including those that require adjuvant therapy. So since then, in the United States, this is the recommended therapy for patients with enucleated retinoblastoma. The ARIA of 321 is the treatment for patients with metastatic retinoblastoma. This is also a trial that requires a comprehensive approach with multiple institutions in the US, with also international participating sites in South America and in Egypt as well. So this trial included 4 cycles of increased cisplatin, cyclophosphamide, and etoposide, with a consolidation with high-dose carboplatin, thiotepa, and etoposide with autologous rescue. And this is where you see the outcomes for patients with a stage 3 and 2 disease, so that is intraocular disease with extraocular extension to the sclera or to the optic nerve. We had a significant improvement in outcomes, close to 85%. Even for a stage 4A, that is patients with extra CNS metastatic disease, the outcome was quite beneficial, with survivals close to 80%. The problem was with stage 4B, which is patients with CNS metastasis, and that showed very poor outcomes, denoting that actually that's the group of patients where future trials should focus. So while we continue to struggle with patients with metastatic and extraocular disease, at least we were able to prove that for patients with extra CNS disease, intensive cisplatin-based therapy and autologous transplant could still cure about 80% of those patients. And now we're focusing on developing trials for CNS disease. So what are the findings from the retinoblastoma study? So we were able to define criteria to risk stratify patients with enucleated retinoblastoma, resulting in excellent outcomes, close to 100%. We define therapy for those with metastatic retinoblastoma with good outcomes for those without CNS disease, and helps us now refocus our attention to patients with metastatic disease to the brain. And so COG has enabled rapid accrual for this relatively rare disease, although we have slower accrual for those with intraocular disease, and we require international accrual for those with metastatic disease. So the next group of patients I would like to discuss is nasopharyngeal carcinoma. So for that, we put together the ARIA of 331, a protocol about 2 decades ago already, that included 3 cycles of cisplatin 5FU as neoadjuvant therapy with concomitant chemoradiation therapy, with radiation therapy, and initially 3 cycles of cisplatin, but the 3rd cycle of cisplatin eventually was removed from therapy because of toxicity and failed feasibility on the study. On the right, you see the accrual. It took about 6 years to accrue about 111 patients on this trial, but the outcomes were quite favorable, with overall event-free survival for the entire group of patients close to 85%. But obviously with significant toxicity related to cisplatin and radiation therapy. One interesting observation that we had already noticed in individual institution-based studies is the unique epidemiology of nasopharyngeal carcinoma, also in the United States. As you know, it has been very well documented. It's associated with EPV and its increased incidence in the Mediterranean basin. Here in the United States, what we saw is obviously already documented a predominance of males, about 14 median years of age. But then what was interesting is the racial disparities, with about 50% of these patients being Black or African Americans. And so for us in the United States, nasopharyngeal carcinoma is a tumor that has a higher incidence among the African American population. So during this time, the nasopharyngeal carcinoma field in adults has also shown, given the ability of actually do randomized trials, the superior outcomes of the gemcitabine cisplatin over the 5FU cisplatin. This has been shown in patients with upfront therapy as well as relapse therapy, as you very well know. And this has informed the next trial for the children's oncology group. So in summary, we had excellent outcomes in children, including patients with distant metastases. It was a group of only 7 patients, but with a 57% event-free survival. We continue to see significant radiation and cisplatin-related toxicities with ototoxicity, xerostomia, dental problems, trismus, hypothyroidism, which made us think about how to design future trials, considering excellent outcomes, trying to define outcomes that actually include decrease in long-term toxicity. And so based on the adult data that we are seeing right now, one of the main strategies that we're going to be studying is the addition of immunotherapy to allow precisely radiation reduction with the goal of preserving or improving outcomes. And this is basically the trial that we have right now, ARIA 2221, with the primary objective of evaluating the safety and feasibility of a cisplatin gemcitabine instead of 5-FU, cisplatin gemcitabine and nivolumab, a checkpoint inhibitor in children with nasopharyngeal carcinoma through determination of CTCAE grades 3 or 4 and immune-related adverse events. And so secondary objectives in this study are response and event-free survival. We have several exploratory studies using blood, tissue, and stool for EBV quantification and microbiome, long-term checkpoint inhibitor toxicities, and response by MRI versus PET as one of the objectives as well, including patient-reported outcomes. This is a single-arm study, although it just stratifies patients based on response for the dose of radiation therapy to receive. And so, we have decreased the dose from about 59.4 to 54 grays, and hopefully in subsequent trials, we'll be able to continue to decrease. As you know, among adults, the dose of radiation therapy can be as high as 70. And so hopefully this next trial will allow us to start that process of incorporating immunotherapy and start decreasing radiation therapy. So the next study we'd like to share in terms of initiatives that we're conducting in the Children's Oncology Group is pulmonary blastoma, an extremely rare malignancy, less than 0.25 cases per million. And so we expect about 19 cases in the United States. So you're very well familiarized with pleuropulmonary blastoma. It's the most common lung cancer of infancy and early childhood. The majority of patients are younger than 7 years of age, is genetically linked to DICER1. So 100% of PPVs have somatic mutation. More than 70% of patients have germline mutations of DICER1, and there are no prior prospective therapeutic trials. You know, the current standard of care in type 1, which is the cystic form, is usually resection. In some cases, chemotherapy has been given depending on pathology. Types 2 and 3, so that's the mixed cystic solid and the solid, is resection chemotherapy. And for those with type 3, usually radiation therapy. The outcomes for type 1 and type 1R, which is the one regressed, are excellent with event-free survival and overall survival close to 90 to 100%. Type 2s are still associated with good favorable outcome in about 60 to 70% of survival. The problem usually, as you very well know, is type 3 PPV. Usually these are patients at higher risk of CNS metastasis and progression, and outcomes are well below 30 to 40%. So with that, putting that together, we developed the ARIA 2131 with the primary objective of determining whether a camptothecin-based consolidation and maintenance regimen could improve outcomes compared to historical controls using the PPV registry that has very well curated data for patients with types 2 and 3, with secondary aims related to the overall response rate and the outcomes of patients with type 1. The protocol is currently in development. We hope to open it in the next year. And finally, adrenocortical carcinoma, another very rare disease, less than 20 cases per year in the United States. For AR-0332, that's a trial that was developed again about 2 decades ago. We tried to define what would be the standard of care for these patients. So we had a stage-based approach or risk-based approach. Stage 1, small tumors completely resected, surgery alone. Stage 2, larger tumors also completely resected. The study was designed to test the role and impact of retroperitoneal lymph node dissection alone with no adjuvant therapy. The stage 3, which was patients with residual tumor, spillage, and resectable, or patients with regional disease like nodal disease, the treatment was surgery and chemotherapy with a cisplatin-based therapy and mitotain. And stage 4, metastatic disease, the same, surgery whenever possible, and mitotain and cisplatin-based therapy. So this is kind of in one slide, a summary of the study. So it took a while to enroll and complete a single-arm trial, and it was done in conjunction with institutions in southern Brazil, where adrenocortical carcinoma is endemic, if you wish, due to a unique mutation, the 337H p53 mutation. So it was activated in 2006. You can see the moment that the trial started with international collaborations, how accrual accelerated, and about 1/3 of all the patients were accrued in 2 institutions in Brazil. You see the outcomes. I will just share the table because the figures are a little more complex to read, but basically excellent outcomes for patients with a stage 1 disease, small tumors completely resected, with an event-free survival of 86%, overall survival of 95%. Lower outcomes for the stage 2, so larger tumors completely resected with retroperitoneal lymph node dissection, event-free survival of 53%, but I want to point to the fact that the overall survival was close to 80%, meaning that most of these kids were salvaged with chemotherapy. What was interesting is for stage 3 patients, the outcome actually was better than stage 2 patients. So these are patients, usually most of them, that had spillage, and so for those, chemotherapy was able to salvage most of those with an event-free survival of 81%. And obviously, stage 4 patients were those with the worst outcome, with an event-free survival of 7%. So basically here, we were able to kind of document the role of chemotherapy and mitotain-based therapy for these patients, but we were able to show that it is an extremely toxic regimen. And so another study that we're currently conducting is about methylation profiling. I want to share some of the early data that basically shows that with methylation profiling, we may be able to optimize staging and risk assignment for these patients. So this is a study done with historical data from the International Registry and some COG studies. But basically what I want to show as preliminary that methylation subtype may be even a better predictor than stage. So we are now validating with the AR-0332 specimens. I'm telling you that the analysis has been complete, and we have a manuscript ready for the complete combination, but basically a combined risk stratification using histology and methylation profiling is quite powerful. So we have other studies within COG, and I just show here one snapshot of how we try to reroute some of the rare cancers into some of the clinical trials for novel therapies based on molecular characterization. And many of the rare tumors have been enrolled on the larotrectinib clinical trial. And you see here some of the results, but this is not surprising to any of you because of the responses of NTRK mutated tumors to larotrectinib. So the lessons learned is that COG has enabled successful study of several very rare malignancies. The international collaborations, the international accrual has been critical to the success of those studies. And then that even in our most common rare tumors, no randomized studies have been conducted because of those same limitations. So with novel agents, we need to identify highly active agents to generate better opportunities for clinical trials. And so in 1 minute, because I see that I am a little over time, in just basically 1 minute, I want to share some of the initiatives that I believe that Dr. Kralo has discussed before me. And this is not a surprise to any of you. I think that Dr. Sam Volchenbaum was here in India a couple of weeks ago. So many of you may have already seen this data about the initiative that he's leading through the University of Chicago on the data for common good. And it's basically an initiative that started about 10 to 15 years ago with a pediatric cancer data commons with multiple initiatives coming together like INRG, the International Neuroblastoma Risk Group initiative that basically started that global data collection. So it grew over the years to become now the data for common good that has several ingredients for success. You know, the community engagement, the robust data governance, interoperable data standards with a lot of curating of the data and optimizing the common data elements, high quality research and real world data, because most of the data comes from clinical trials, processing and harmonization pipeline, data discovery and management platform, and then usually a very user-friendly cohort discovery. This is basically the data for common good, the pediatric data, cancer data commons that probably started this process with the world's largest harmonized, publicly accessible clinical pediatric oncology data set. But in more recent years, other diseases have come to join this initiative, like monogenic epilepsies, other rare diseases, monogenic diabetes, and even other initiatives like gun violence and others. This is a snapshot of the different groups in rare cancers. We have retinoblastoma, the global reach, and esophageal carcinoma, novel, and others that would be considered part of the rare tumor categories like CHIC for liver tumors or MAGIC for germ cell tumors. And this is now a global initiative with many institutions in North America, clinical trial organizations in South America, in Europe, and Asia being part of this initiative to continue to pull data together to generate that common good, that big data group. And I know that there are conversations to include as well INFOC and others in India as well. So I want to stop here acknowledging many people that have contributed to that, especially Ted Letts, the current rare tumor chair at COG, the vice chairs, Chris Schultz and Murali, the COG leadership over the years, the entire rare tumor committee, and obviously Sam Bolchenbaum and the team for data for common good. So thank you so much. Happy to answer any questions a little bit later. Thank you.
Thank you, Carlos, for that talk. I believe you've also joined us online, but I would kindly request you to stay on and we'll move on to the last topic. So expert insights, the way forward for pediatric oncology. And I'd like to invite Shripad sir to deliver this lecture.
Thank you. Thank you, Carlos, for being there. And thank you, Nikhil, also for being online. And we miss you all here. We would have loved to host you here maybe next time. Can I have the next slide, please? Thank you. So I have been given a really broad topic and just thoughts of what this is a talk in the making, though it's not complete. I'll present some of the views here. So, not our Maya, but some other Maya Angelou, who had spoken some time back that I have great respect for the past, because if you don't know where you come from, you don't know where you're going. So let us have, what we have learned over the years and how we can improve the outcomes of children with cancer. So here we have seen, actually, we have heard again and again that over the years, the outcome of pediatric cancer has definitely improved. And especially if you consider the early 70s and now there has been definitely a lot of improvement. But again, like in adult cancers, basically like pancreatic cancer and all, there are certain pediatric cancers also where the outcome has really not changed over the years. And just now you heard Carlos mention that especially stage 4 cancers, I don't know, though we show a lot of improvement in other cancers, the real cancers, the stage 4 cancers, the metastatic cancers, whether we have really improved over the years. And definitely, I would like to mention here that we have to give a lot of credit to this gentleman because he was the one who really gave us the first drug, the methotrexate, and even many antibiotics. And it was told that he even gave actually the mercaptopurine, which later, was really discovered by, the Nobel Award-winning scientist. But definitely, he contributed a lot to this what we are doing now in chemotherapy. So, I thought I'll just mention this Dr. Subbarao, who basically was never even given a Padma Shri for that matter. But the most important thing I think in pediatric cancer is to create awareness because still, I know many people still feel that cancer is a disease of adults. And, people ask me when they ask me what do you do, and if I say I'm a pediatric oncologist, they ask me whether pediatric children do develop cancer. But definitely we have to create that awareness that, basically a lot of the people still don't know. So we have to create that awareness about pediatric cancer, create the awareness that pediatric cancer is highly curable. And very importantly, there are still a lot of misconceptions, especially in India, that it's a curse that yesterday also somebody mentioned, that it's a curse that, children develop cancer, or basically it's a communicable disease. They don't want other children to be with them. So a lot of misconceptions which have to improve. And, just now also you heard, in 1971, the United States declared war on cancer. And later, even now, that the President Biden, as well as the President, our Prime Minister Modi, actually did this quad moonshot. And we talk a lot about cancer moonshot, but I think especially in a country like India, earth shots are important. And very importantly, editorial sometime back in Nature, he told that it's sometimes very difficult to, earth shots are really harder than moonshots. And that's very important to know what we are and where we are before talking about the moonshots. And what is more important, actually, in India, for example, if you see, and we are talking again and again, and last few years this has been discussed, very importantly, is we don't even know what is the amount of the magnitude of disease in the country. So I think the pediatric cancer registries are very important. And we have to know the enemy because we have to even allocate the resources accordingly. So unless we know what is the amount of disease there in the country, where they are, where they come from, will not be able to give the resources properly or to even develop the infrastructure to take care of this. So in India, one of the things which is the cancer registry is very important. And sometime back, the ICMR, the Prashant Mathur and the team did this very important study to assess the childhood cancer care services in India. And they found that most of the services, and we all know that already, that most of the services are in the urban areas and in the bigger institutions, and that also many times in private institutions. And we have to develop the pediatric cancer care even in the government hospitals, and very importantly, the medical colleges, because there the pediatric cancer care is really lacking. And the registry data, Venkat, I don't know, Venkat is here. He was here yesterday, I think he's not here, but Venkat actually did a very, very important work, that is the development of a population-based pediatric cancer registry in Chennai. And I think this work needs to be taken all over the country, and through the pediatric hemonc chapter as well as in POC, with the help of the cancer registries of the country. I think we'll have to take this as a big project for all these three together, and to make sure that every child with cancer gets registered on the cancer registry data, so that we know where the patients are, what the disease is. Because very importantly, actually, when we talk about the the number of patients, I think we are inflating the numbers. Personally, I don't think there are 55,000 childhood cancer, which this number is shown many times, whether there are that much. Because what will happen is if you overinflate also, we are training, because the second part of it is, if you know what the numbers are, you know how much to train, what are the numbers that to require, because the second part of it is education. because unless you have trained people, you cannot take care of the pediatric cancer patients. And I remember I was a trained adult oncologist, but I changed to pediatric oncology because that time there was no really or very few trained pediatric oncologists in the country. So that time I got, but now there are really many pediatric hem-onc uh, doctors being trained, pediatric oncology doctors being trained, and also basically many people who have done the board certifications or training from UK are coming back. So from outside also people are coming. So there's a lot of interest in this field, lot of trained people in this field, and a lot of FNBs also being trained there. So it's very important to know what is the magnitude of the problem. The other area that, through the pediatric hem-onc chapter, we are training even the nurses, because that's very important to train even nurses, because they really take care of many of our patients. And through again, the pediatric hem-onc chapter, through the national training program in pediatric oncology and pediatric hematology, we are training even the general practitioners and the pediatricians out there, so that they become aware of the disease, aware of what to do, at least the basic things, so that the patients will be referred early and referred properly to these centers. So this is what I wanted to show. We think that there are not many trained people, but if you see here, nearly 70 doctors are being trained in the field of pediatric hematology and pediatric hem-onc every year. So it's a big number, and we have to make sure that all of them get good jobs, so that how much more to increase the number, how much more, all that will depend if we know exactly what is the load of disease in the country. But along with that, actually, though we train a lot of pediatricians in the field of pediatric hem-onc and pediatric oncology, I think this is an area where I don't know, Sajid was here. Sajid, were here somewhere? We have very few trained pediatric oncosurgeons actually, and that's why a lot of patients are being referred to centers like Tata and all for surgeries, because there are really few trained pediatric oncosurgeons. Even in Tata, we are really hunting for a second surgeon, because there's not many trained people out there. But we have to train more, I think, pediatric oncosurgeons. We need more pediatric critical care specialists, palliative care specialists, infectious diseases, endocrinology, cancer genetics. So there are many other areas which need to be looked into and developed, because they all together we treat children with cancer. So just training pediatric oncologists will not be good enough. And I think somehow through the Indian Academy of Pediatrics, we'll have to make sure that the other things are also trained in equal numbers. Then comes the service, actually, that's very important. And very, very importantly, actually, over the last years, because we have now increased people who are trained in this field of pediatric oncology, as well as the government basically support that we are getting. So through various government schemes, through various CSR funding that is available now, and even the volunteers, the various NGOs, there are many, many more pediatric cancer units which have developed over the years, and many centers are now treating children with cancer. So unlike initially very few centers, and those also only the metro areas were there, now there are many more pediatric cancer units across the country, and they are getting a lot of supports through various schemes as well as through CSR funding. But very importantly, this is another part of the thing that the pediatric hem-onc chapter did and is still doing. Actually, Neeta is doing at present is a global mapping, that is knowing all the centers that are treating pediatric patients, so that there's a global mapping, so the patients can be referred to an appropriate center for treatment. So global mapping is important, and it's an ongoing task, which I think Neeta and her team is, and the IP chapter is doing at present. But very importantly, actually, we wrote a letter as part of as being a chairperson of the pediatric hem-onc chapter. We had written to all the ministers as well as the government officials that actually there should be a pediatric cancer unit in every medical college, because there is where the training is, and that's where patients will get good care also because of the infrastructure. So this is still in progress, and it would be very important for even employment for the so many pediatric hemong people that are getting trained. But at present, this is still in progress. We have written to them. I think we need to follow it up on a more aggressive basis. So what it is actually service when you tell, there's various ways to treat children with cancer. And one of the most important thing is basically for a country like India, actually where we have seen that we are losing nowadays many more patients of toxicity rather than basically relapses. It becomes very important to set up a line and to demarcate how aggressive one needs to be. When you see a foreign protocol like the LMB or the hyper-CVAT protocol or the flag IDA protocol, people want to use that immediately because the outcomes are good. But ultimately, if you're losing more patients of toxicity, that whether 2-4% improvement which is there of a protocol, whether you get that. So I think we have to focus on what will be more important and the risk refinement, whether we should minimize the toxic deaths or minimize the relapses. So both ways we want to do that. So we have to make sure the risk stratification becomes again more important for us to do, so that we always feel that the molecular characterization, the cytogenetics, these are luxuries and they are not required. But I think if you risk stratify patients properly upfront, then you can treat the tailor the treatment so that those patients who don't need aggressive treatment, you can decrease the treatment and importantly decrease the toxic deaths. So I think it's very important to make sure that there are no increased toxic deaths just to trying to get more decrease in the relapses. And this is important actually for the West. When we talk about science, we always talk that the treatment outcome depends on the treatment given, the responses, the pharmacogenetics, the seed, the soil, so the biology of the tumor. But for a country like India, we have seen that along with this, we have to take care of the nutrition, the supportive care, and even the drug compliance and the drug quality. And this becomes very important because when you read, see, 90% of the data or this thing comes from the West, and there you'll not be getting, this thing when you talk about biology and all, we don't talk about this in many of the Western protocols. But for us, I think it's going to be more important in this. For example, just to see, actually, some time back, we analyzed the induction that, when we had done that out of the 18 patients that died in ALL induction, very importantly, 14 of those which died of sepsis actually had albumin less than 2.2. So they had very low albumin, whereas the patients who had more than 3 albumin, there were only 2 deaths, and out of which one was because of sepsis and one was because of the hemorrhage. So very importantly, low albumin may be a causative factor in induction deaths in toxicity in our group of patients. And we had hypothesized at that time that maybe the low albumin in the presence of low platelets and low ANC it propagates sepsis. And this may be because there's basically edema of the gut and there's migration of the bacteria from the gut out. And with low ANC, it causes sepsis. So maybe that may be an important factor which we'll have to look into. And maybe this is our problem. And somebody, I always tell my residents, we have told them that they should do a proper study of this and see whether that's a causative factor, and we can take care of it by making sure by infusing albumin and making sure the albumin is high in this group of patients. The other thing, this is a very paper which is often quoted. This group in Malaysia doubled the outcome in patients with AML by not changing the protocol, but by just improving the supportive care. So very importantly, just when the outcomes are low, it's not a knee-jerk reaction is to change the protocol in this thing, but very importantly, along with the treatment protocol, supportive care becomes very important. And Nikhil mentioned in that, what does supportive care mean is a very, very big term, but we have to sit with that and see how we can improve the supportive care along with the treatment protocol, and maybe that will help us improve the outcome, and this becomes more important for a developing country like India. And this is again our work, which has been often quoted and reported everywhere, that out of the 9 brands of L-asparagines that we had, and one, the original one, the Lunes from Japan, the original one, all the Indian brands had much lower value as compared to, the levels as compared to the one which was the original. So very importantly, and when this L-asparagine becomes one of the important drugs that we use in our protocol, whether, if you have a low quality drug, how I feel it will affect the patients and the outcome of the patient. So this is very important, that not only the biology, the quality of drugs, the nutrition, all that becomes as important, the supportive care, all becomes as important as the treatment protocol. And this is what actually, chemotherapeutic drugs, most of the drugs that we use in pediatric group are the original, the old drugs, which are really low cost. And now the government of India, with this, the price controlling that it has brought down the cost of drugs to such a level that it becomes difficult for a company to make profit out of it. And that's why many of the companies are now stopping making the pediatric drugs, including donomycin, vincristine, all these, even holoxan, the ifosfamide, all these drugs were becoming out of stock in between, and they keep on going out of stock. And that's one way, important thing that we have to keep in mind, that if you want to have good drugs, I think the government should allow these companies to make at least some profit, a good number of profit, because no company is there for social reasons. It has to make its own profits also, but very importantly, that also will affect the not only the availability, but the quality of drugs also gets severely affected when we don't allow them. The drugs price is brought to such an extent that it becomes difficult for the company to remain in the market. So we have done a lot of, basically advances are there at Tata. We have the robots to do the surgeries. Yesterday you heard Dr. Siddharth mentioning about the proton therapy and what all we are going to do with that. There are a lot of targeted therapies available. The cars are there. Gaurav showed the CAR T that we have produced. So all the advances are there, but you have to keep in mind that we are in India. So we have a group of patients who can afford the best. They can go all over the world also. But at the same time, there are people who cannot afford even the standard of care. So we have to keep that in mind. And that's one thing actually. I work in both adult and pediatric areas. So the metronomic therapies, the drug repurposing is an area of very great interest to me, and I've been working on that. And we have used it in acute myeloid leukemia. We have used it in acute promyelocytic leukemia. We had a protocol for patients with relapse recurrent Ewing sarcoma. For example, this is one patient with such an excellent response. So definitely there are patients who respond very well to such type of treatment. And I feel especially patients with stage 3 disease, who have a lot of micrometastasis. The ALL, we did a maintenance therapy, it worked very well. But I think that type of treatment, even for solid tumors, would be required, especially for the advanced patients and especially stage 4 patients. Actually, you saw the data even now, what Carlos showed. Stage 4 patients still don't do well, and that's because I think the success that we had with our other patients, we had the success that we get for treating stage 1 to 3 patients, we use the same strategy for stage 4 patients, and we have seen that it miserably fails, because I think stage 4 is disease not of the tumor itself, but the microenvironment, and it's very important not to over-treat or overkill the patients with giving the high-dose therapies, but we have to take care of, basically the microenvironment, and very importantly, the immunity also, and that's why now, with the addition of immunotherapies to our chemotherapies, it is changing the way the outcomes of these patients, but it needs more work. And that's why, especially in country like India, where we get more percentage of patients who come with stage 4 disease, I think it's very important to look at stage 4 in a different way also. And not only that, even the standard therapies which are there, there's a lot of areas where we can work on. For example, this is the case of raspberry case, where the regulatory trial did the dose of 0.15, per milligram per kg for 5 to 7 days, and that becomes the standard of care. So it really becomes very expensive to give respiratory care. But we have seen that we have given only one dose, one vial, which is 1.5 milligram to even a 60 and 80 kg patient, but it works well, So really, what is the ideal dose? Whether the dose which has been published, it's the right dose, and this I'm giving example of just one, molecule, but there are so many other things where we can work on it. And this was the one which we did the low dose immunotherapy, which along with the metronomic therapies, when we added, we saw that low dose works really well. And now yesterday, Sham showed the data of even Hodgkin's disease, where we use the low dose immunotherapies, especially Manju is here, the cost effectiveness, whether we need the whole, the standard dose or low dose may be as good and maybe better sometimes if you use it along with other therapies. So that's one area where we need to work on. And for that, actually, and this thing, especially for a country like India, where supportive care is very important and expensive, sometimes, this type of the antibody therapies may be much cheaper in the long run and more doable also. And this, looks very simple now, but this slide I've been showing for the last 8 years now, and I think that's going to be the future where we are going to really decrease the intensity of chemotherapies and take help of antibodies and other thing. And here again, there's off-the-shelf versus personalized treatment. There's an antibody versus a CAR T cell. I think for a country like India, off-the-shelf will be much cheaper and much easier to do as compared to CAR T cell, though we may need it for patients who advance, who have relapses. But in the initial phases, I think we'll get away by decreasing the intensity of therapy, but adding this the newer molecules like antibodies. And for that, we need to do a lot of trials. As we all know, pediatric cancer has improved because outcomes have improved because of trials, which again, Dr. Carlos just showed. So we have in POG here, Dr. Ramandeep is here, but the INPOG as well as the National Cancer Grid, we have, basically areas where we can work, do a lot of locally relevant research. We started with MCP 841 protocol years back, where we showed we improved the outcomes of ALL at that time. But now recently, we also did the large ICICL trial, which had more than 2,500 patients on that. And again, we showed that we could improve the outcome of these patients with ALL, and at the same time, we even do randomized large trials there. And now this is going to be done in nearly 21 centers across the country. So what we need is basically we need a lot of people to treat children with cancer. I'll not touch the data commons because definitely there's one thing that India will need where hardly 5, 10, 15% of our patients are on trials. There's a lot of individual patient data, which if it comes on data commons, I think it will give a lot of real world data to see how we can improve the outcomes of a patient with cancer. So ultimately, I think every child with cancer deserves the best chance at cure. And we have to keep in mind that it's not simple when we always say that childhood cancers are highly curable. We know that they are curable only if they're diagnosed early, diagnosed properly. That's very, very important, and treated appropriately. And lastly, the universal prayer that, that every child with cancer be cured. So thank you very much for your.
Banavli, for that wonderful session on LMIC perspective on pediatric cancers and the way forward. Now the session is open for comments or any questions.
Right from how data should be analyzed, what are the real world perspectives, what is our context, what is the ARIA framework where you can do risk adapted management. So it was a very wonderful session, and I would like to invite questions. So Dr. Carlos is here and Dr. Nikhil Bhakta, both of them are online. So questions for them. Yeah, go ahead, Sham.
I have a question for Krelo. Oh. Yeah. Hi. Wonderful talk. My question is on your Bayesian theorem.
So, two questions. You were talking about arriving at a cutoff, say 80%, 90% of that sorts. If you're trying to arrive at it and if you're successful at it or whatever, how different is it from the frequentist approach where we always try to, reach the 0.05? I mean, is this not a step going back and trying to arrive at a cutoff and rather just leaving at the probability of it? Is that not the whole point of Bayesian? That is my first question.
Well, there are Bayesians who say that you get your posterior probability and you go with the one that wins, just anything above 50%. I think that even in the frequentist world where we chose 0.05, there is no good evidence that why it's 0.05. I think what we have to examine is if you're going to do trials in a sequence, what's the right p-value to have so you can make progress? You can imagine that if you chose a very small p-value, it would take you 20 years to do a trial, but you're really sure you got the right answer. And maybe you need to, and I think those two papers we saw on what is the right strategy, I think those we have to re-examine from a strategic point of view what the right thing to do is. I don't think we know yet, and I think we have to do that.
Great. So my the second question is on priors. So when you're choosing a prior, heavily dependent on your historical data, probably the data coming out of the study, which is ongoing. So you could either have an informative or a non-informative prior, if I'm getting this right. My question is, when we look at a paper, when we read a paper, how do we know when one is using a Bayesian approach, how much of this prior is influencing the outcomes of that study? I mean, can the data not speak for itself, and whether the study itself was a success or a failure, or is it a success because we have incorporated prior into it? I mean, are there...
Right, you're saying, because I know that one of the reasons that we've used non-informative priors, and we do that a lot with our funding agency, is that informative priors, you're right, they can give you the wrong idea just because you put too much weight around the answer that you love. So we do a lot of non-informative prior work. There's also some other, like the Bayesian information criteria, where you can see, has the amount of information you gathered from the trial, does it overwhelm that prior information too? I did not talk about that, but that's a pretty technical thing that, and a well-reported trial tells you what those criteria
So they're supposed to say both without considering the prior and after considering the prior. Is there some reporting guidelines on Bayesian outcomes?
Well, actually, there right now are none. I mean, when I've written them up, I don't say what the analysis would have been like had I done it like a frequentist or had I done it like a Bayesian. I just say, I do it like a Bayesian. Here's my prior, and here's how much information was in there before, and here's how much information was added when I look at the BIC to tell me how much I actually gained over what the prior was. But that is a reporting habit, which I think we all as statisticians should take care of. As you're right, how much did you actually overwhelm your prior with the information you gather?
But that's still not there in the sense.
I think there's a lot of people who don't do it. There are some of us who feel obliged to do it. Okay. And I do want to get people to start looking at reporting guidelines. It's an important thing.
Thank you. That's all.
I'm just so happy to see somebody speak out against 0.05. It's like a religious faith.
Oh, yes.
And for decades, I've been very clear. So I really appreciate it. And again, this board, it's not going to change anything. People go crazy. But then probably be better. People can hear me now. It's just like, people go crazy about getting that 0.05 mark. If it's 0.04, they're thrilled. If it's 0.06, they think the whole thing is a waste. But I really am, so thanks. Appreciate it. Second question, I had for Carlos. So it's, I think he's still on link, yes. So the COG CCDI trial, molecular sampling. So as far as I'm aware, that was limited to the US. And I think that collection for molecular samples is still underway by COG. I mean, Doug Hawkins would know better, but that's my understanding. Is it open to have satellites in other countries or regions? Because that is one thing, we have like an abundance of patients and samples, but I'm not sure we have clear-cut collaborative links. And as you know, sending anything that's DNA-based country to country is always a challenge. I think the picture may be frozen.
Carlos, you there? No, that's it. Just
I'll take the answer.
okay, but
Hey,
may-
Vic, sorry, I lost you for, I
sorry.
lost connection here for a second, so.
Oh,
Yes.
okay.
So thank you so much, Vic. This is yeah, this is a great question. So yes, I think that as far as I know, when Mark to these molecular characterization studies are just limited to the COG institutions. You know, COG has explored very actively these international collaborations, and actually the retinoblastoma study for a while was open as well in India. But I think for the molecular studies, that has been a little bit more challenging. It has to be study specific. So for the adrenocortical carcinoma that was open in Brazil, or the retinoblastoma, some of those, yes, that included these biology specimens across countries. But that's a very highly regulated area, as you very well know. And I think that addressing these challenges is going to be critical. Yeah.
But Carlos, but Dr. Banavli, Sripad here. Sorry, can you see here?
Hey,
Hello?
Sripad. Yes.
Yeah, so
Yes, I can see you. I can see you, Sripad. Yeah.
so basically, if you can still collect the molecular samples, and we can do the test here. I mean, India or many other countries will now be equally equipped to do all the molecular tests. So why should not it be there? Doesn't have to go every.
Definitely.
But very, very importantly, we can do so.
Yeah. Yeah. No. That.
Yeah.
Yes. Definitely. Definitely. I think I'm just talking only about how the COG study is a structure. As you know, COG has different regulations, different structures. That's not to say that the international collaborations are not critical at St. Jude, and Nikhil is here. Maybe you may want to comment as well on that. Nikhil is leading an effort that includes as well institutions in India that is trying to overcome this. That's the DVS study that definitely is going to be probably a model to follow. Definitely. There are many other ways of sharing samples. We can do bedside sequencing and use the cloud systems to actually analyze some of these samples and share them. So there are many other ways to advance in this.
So Mark, I had one Mark here. Sorry. Yeah. So this thing, that say I do adult oncology also, and we have 8,000 patients to get that 0.05 level, So I really feel whether it's, whether it's clinically any relevance to have 8,000 patients to get that and then call it standard of care and change of practice and all. So very importantly, I think we have to change this 0.05. You are absolutely right. It's no longer going to be, no, you are going to see, say, 200 patients. If the difference is seen in 200 patients, it's important. To get 8,000 patients to get 0.05 and then use it as standard of care really doesn't make sense. But, I had one question for Kathleen. So, the PHAP program is.
Sir, can I just continue on that? Because you just said 0.05. So I'm just asking, so if you are refuting 0.05, what is the, are you removing the guardrails or you want to tell everyone that you do Bayesian? 0.05, which is said. And it's a
But
very
I think we have,
pragmatic question that I was asking.
I think that we all got tied to this 0.05 because somebody wrote a guidance back in 1972 about what the right thing to do was. I think people are looking now at why that's a good value, and that's why I put those papers up there that said at some point you have to make a go no go decision that has to be made on some criteria, and it's trying to figure out what the maximum, how you optimize that so you get the easiest path forward, how you can make progress. Do you do one giant study? Do you do lots of small studies and hope the small studies get you to the goal of a cure faster, just because you're testing more and more things? It's hard to make a lot of progress if all you can do is test one thing over 6 years. You want to be able to test more. So that's why I think we have to look at that interplay of, I'm really sure I have an advance with a 0.05, or am I less sure about any one single advance, and then move the whole field forward by occasionally making a mistake, but examining enough things to bring a cure.
The effect size is what you're mentioning. We should focus more on the effect size rather than the significance.
Well, we should look and see. I don't want to go too deep into it, but we should look and see to me, it's the number of studies you can do, it's the number of interventions you can try, because you're more likely to identify an effective thing if you look at more things. And how do you look at more things? You do trials that are smaller and take more risks at accepting a therapy, knowing that you aren't as sure, but maybe that's what.
Thank you. Thank you, sir. Sorry, sir, for
Sir,
interrupting.
So Kathleen, one with this thing, so BHAP program we are doing well, but as you know, for the standard of care after giving Blina is to take them for transplant. For a country like India, where transplant is not that feasible for various reasons, logistics, infrastructure, all that, whether the program can allow us to do something different. For example, we, after giving those 2 cycles, whether we can give for 1 week, for every month, for next year or something, some different way to see that we can get away with transplant, but you use your drug effectively. So, those investigator-initiated trials, if you can give us, I think it will really improve the effectiveness of the molecule itself and improve the outcomes also.
Thank you for the question. I don't think we disagree. I think we're recognizing that this is a phased approach. And one of the things that we're trying to do with the clinical data is to specifically address some of the transplant-related questions, because I think there's the Locatelli experience where transplant is really, really important. There's some of the experience from the BLAST studies where whether patients went to transplant or not didn't seem to be as impactful on survival. And I think acknowledging what the standard of care is and acknowledging what proportion of patients where transplant is not going to be an option is really important. In the clinical data that we're collecting, we're specifically stratifying within each of the indications by patients went to transplant or not, because I think some of the important questions are going to be like, can we identify which of those patients we really should be focusing on? And then I think that this is also going to give us a lot of good baseline data to then ask some of those more intensive questions, because I do think we need to design real clinical studies to answer some of these outstanding questions that really wouldn't be pertinent based on the standard of care in the United States that desperately need to be answered. And I think, we all have questions about duration of Blina, timing of Blina. We acknowledge that within the drug donation program, we exist in kind of a confined space. And I think it's going to be really hard to answer some of those questions based on the conditions of the drug donation program. And I think the only way that we're going to be able to move beyond that and have the flexibility to ask some of these really critical clinical questions is by investigating some of these subsequent strategies to improve access. Because I don't think the drug donation program is an end-all be-all. I think it's a really helpful tool at the very beginning, but it's not it's not going to help us answer some of these questions that impact how we deliver care.
Now that you know we can use the drug safely and effectively, please, that may be the next step, like you told,
Yeah, I think I think we're all, Dr. Cheatham especially, always reminds me that those are the next questions. So I appreciate that.
My question is to Nikhil Bhakta. So this is regarding going by what you have presented for the adaptive guidelines. So already there are like many experts who are overlapping with your group and also working in individual countries like in India itself, the ICCI, NCG, trying to simultaneously produce adapted guidelines. So my question is the duplication of work and then, eventually maybe one of the documents or these documents may be redundant for the person finally, when the clinician is using them, the practitioners and the providers, especially for survivorship. So there's a lot of effort and duplication in that space. And then the other aspect is when there are already like pre-existing guidelines in individual countries. So in what way are you working to make them more, implementable for the people on the ground when they, would want to go and use these guidelines?
So, Nikhil, before you answer, it's Maya here. Hi. I just want
Hey,
to
Maya.
get back on Ram's question. And that was that, I mean, this has taken a huge, tremendous amount of work. And it's always very inspiring to see how much work and how much, thought has gone into the ARIA guide. So I'm sure that every disease group or every topic that you choose takes maybe from start to finish around 2 years to do. How sustainable do you think this is? And whenever there's new evidence, how do you plan to update that?
Yeah, no, thank you for all of the questions. So I think first of all, it's important to recognize that the concept of adaptation and the development of guidelines themselves is a relatively new topic. So what we see is great heterogeneity in terms of how people approach the question of adaptation. A lot of times in many countries, the guidelines that are produced, and India is at the forefront of some of the most stringent and some of the most thoughtful protocols that have been developed through INFOG. But most of the guidelines in many countries are either just adoptions of existing high-income country, or they're adoptions with some just tweaks that they make here and there, but nobody knows whether they're actually going to be successful or not. So utilizing the best evidence that you can, incorporating consensus-based methodologies with evidence synthesis and using the power of context experts, in other words, people who can actually speak to whether it's feasible, is a relatively new concept in the guidelines field. In fact, no one else has actually taken this same approach to scale. And so to think about why we need ARIA and why we need to, first of all, I think we all recognize that redundancy is a waste of everyone's time. So the more that we can work collaboratively together to reduce that is always a benefit, I think, to all of us, because we'd rather spend more time studying and developing new implementation models or studying the adherence or the effectiveness of the guidelines themselves, and then actually coming back and improving them. But the other component is most countries usually have a set amount of time when they need to review and then update. And what we're doing right now is actually working with the WHO, because the WHO has a guidelines review committee. They produce their own guidelines. And what happens is that for many of the communicable diseases, the countries will just accept or adopt guidelines from the WHO that are produced and reviewed. But there's two problems. One, that there's no mechanism for the WHO currently to review external guidelines. And two, the WHO only produces about 250 to 300 guidelines per year. Now, if you look at the ICD-11, there's about 11,000 or 12,000 different diseases. So there's no way that the WHO is going to be able to catch up with that, which means that they're going to have to start looking at external groups to do this. And when they started looking at how external groups are producing guidelines, nobody is doing this at a global level, meaning incorporating representation from all 6 WHO regions, all income levels, et cetera. So what we're doing is actually, it sounds simple, it sounds obvious, but what most people don't realize is that as pediatric oncologists, we generally tend to get along and we tend to work well with each other. I think if you look in the adult community, you can see that same camaraderie does not exist. But as a result, what we have is a rare opportunity to show how we can develop guidelines in a novel way. And so you're right, it does take a lot of effort and time, but what we have is a couple of things going for us. One, we have a lot of energy and enthusiasm within our global community to support and develop these. The second is really thanks to Carlos and St. Jude, we've been able to support and grow the ARIA program through our coordinating center that we've developed at St. Jude just in the last couple of years. And so we've been scaling that up as well, which has been really instrumental to developing these processes. And then the third is we really hope that as we start to kind of we spent the last few years developing and actually more or less perfecting this methodology. And so now we hope to be able to scale this at a more rapid speed. So as we get these guidelines moving, we expect that we'll be able to move much faster, but it still will take time and effort till the next few years to really populate all of those 100+ guidelines that I talked about. And then finally, as we do develop this, we are developing processes so that we can think about, are there automated searches so that every X months or years that if we're constantly updating the literature, reviewing the literature and kind of double checking to make sure nothing new has come out. The good thing is that in pediatric cancer, we generally know when there's a major application. And the bad thing about pediatric cancer is that they're very rare for many of the rare tumors. We usually only see major advances only every 5 to 10 years. So as a result, those updates at the higher strata are not as big of an issue. It's more about the adaptations themselves. So we do want to make sure that these are evergreen and are constantly updated with probably more substantive updates every 5 to 6 years.
Thanks everybody, all the speakers. I think this has been a fantastic session where we have seen from traverse the ground from the generation of evidence and to novel ways to do it, to clinical trials, to evaluating that evidence and then implementing it. I think we covered the whole range of topics. But just coming back to one area which I'd like to think about. You talked about INFOG and ISICL, Dr. Banavli was mentioning, and as part of the core group for ISICL from 2012 onwards, when we were very excited that time, we were actually rolling out the first multi-center randomized control trial in the country. And now we have gone on to ISICL 2, and we are also now Hodgkin multi-center trial is going on, and so are many others coming up. So we're really proud of how much we have traveled, how much distance we have covered. But in the global context, we can say that probably the multi-center clinical trial system is just maturing now over here. But it doesn't escape our attention that, like with ISICL 2, for example, we're going to be answering a question which 10 years down the line will probably be redundant. So I think there's a need for us to incorporate many of the things which have been covered in this meeting, including multi-arm trials, etc., which can run simultaneously, to add new interventions on the backbone, use Bayesian modeling to be able to answer questions faster and quicker, because we not only need to catch up, we got a lot of catching up to do before we start innovating and finding newer methods of treating. So if we can get this kind of dialogue going between, I would like people to think about getting this dialogue going and creating the infrastructure for us to move forward from this multi-center randomized control trials, which we just started doing now, to newer methods and methodologies with probably the expertise across the groups to design better trials and faster.
Can I just make one comment build on that, which is obviously mentioning ISICL 2 and the innovations there, but one thing, and I wasn't here for the whole session, but one thing that I do want to congratulate the Indian colleagues on, which I think is incredibly innovative and one that I have not seen in any other context, is ISCAL. The 20+ center implementation study that's being developed in India right now is probably one of the most unique and most impactful ways of trying to establish what is real world practicality for implementation of the standard of care, and the rigor with which it's being done is really exciting. And of course, building on the kind of implementation will at the same quality improvement kind of models, because I mean, you talk to Mayur and the team thinking about just diagnostics and how much effort it takes to go and make sure that each of the centers are actually risk stratifying appropriately. These kinds of studies, not just only the fancy multi-arm Bayesian clinical trials or even the standard clinical trial designs with novel therapeutics, the back to basics, can we implement at scale what is the standard of care in settings that haven't traditionally done it? That is incredibly important. And that design of that study is something that I'm hoping other countries will learn from and be able to implement as well.
For that comment. Thank you for that comment. And I know there are a lot of questions. Dr. Banavali, you'd like to have some words? Yeah.
No, again, Nikhil, again, the same thing here, now again, we are going to do 4,000 patient trial and, which has no, for example, no molecular data, no antibodies are being used. So what, like Gaurav mentioned, for 10 years down the line, how relevant will that? But I think the most important thing that we learned with Icicle is that we can work together, we can treat patients on protocol basis and uniformly across the country, improve the infrastructure, improve the diagnostic. So I think more important than the outcomes and basically the other part, I think we learned a lot doing the Icicle trial. And the same goes with the other INFOG and other trials. What you all have done years back, we are now slowly learning. So I think that's a good this thing. And like you told, pediatric people like to work together. So we are bringing more and more people together. I think that's the best achievement that is to begin with, and later things will fall in place, and we will change, Gaurav, the way, maybe down the line we'll have to change the way the whole treatment will change. I know for ALL, the way we have Icicle, I don't think that's going to be a treatment 10 years down the line as it is, but at least let us work together and slowly will change.
Right, right. So thank you so much, sir. And thank you, Nikhil. Thank you, Dr. Carlos, for staying awake. It's almost midnight in Memphis, I know that. And we very much appreciate your presence here online. And we are sorry we could not get the visa. But yeah, many thanks to you from all of us. On that positive note about implementing what we are doing right now, the trials, and Dr. Banavali and Dr. Nikhil made a very important point. On that positive note, I think we will break for tea, but be back in 10 minutes so that we can start the session. Okay, thank you so much.
Thank you, everyone.
Thank you.
Thank the chairs. Hello, hello.
Hello. Welcome all. So to the last session of this track. So this is on diagnostics and therapeutic advances. So for the chairpersons of this session, I would like to invite my colleagues and seniors, Dr. Nehal Khanna, a professor of radiation oncology, Tata Memorial Center, and Dr. Siddharth Laskar, who's a professor radiation oncology and deputy director academics, Tata Memorial Center. Over to you. Change the schedule. Dr. Nehal.
This is the fourth time Maya is announcing that the mic takes 10 seconds to initiate. So we had a very nice and interesting session on how to implement the evidence and guidelines onto routine practice. Now we'll start with these three talks. The first talk will be by Dr. Jeffrey Orjan. He's from Seattle Children's Hospital. He'll be speaking on the importance of artificial intelligence in pediatric radiation oncology. This will be followed by a talk by Dr. Jifmi. talk on pentech. This is one area which every single radiation oncologist who practices pediatric rad onc must be aware of. It's going to be a very inspiring and a very insightful talk. This will be followed by a talk which I will be giving on advances in pediatric radiation oncology. We welcome Dr. Jeffrey Orjan for his talk, please. Thank you.
All right, thank you for having me. This is my first time in India and it's been very exciting. All right. So I'm a pediatric radiologist at Seattle Children's Hospital in the United States, and there's a little picture at the bottom. I know not everybody's familiar with US geography. It's very top left is where I live. I'm at Seattle Children's Hospital in the University of Washington, which is right next door. And because I'm a radiologist, I'm going to be focused mainly on artificial intelligence and radiology, but that's only kind of an overall guideline. A lot of what we're talking about will apply to any clinical specialty. So I don't have any financial disclosures. Nobody pays me to do anything, sadly. We're going to be discussing some investigational products and techniques, though. So a lot of what I'm talking about is future or investigational. So take everything I say with a grain of salt. So we're going to talk about who I am and an AI overview. A lot of you will be familiar with AI in general. Examples of current use of AI that we're doing right now in radiology, some pitfalls and challenges that AI is involved with as well, and what the future of AI may be. And obviously that is something that is just out of my head, and lots of ideas about what the future of AI might be. So I originally was a pediatrician and studied and trained to be a pediatrician, and quickly learned that I liked computers more than people. So I turned into a pediatric radiologist after several more years of training. I'm not a computer scientist. I don't code these things. I rely on other people to do these for me. I am an end user of software like everybody here, but I consider myself kind of a power user and fell into the role of a physician scientist doing projects in AI, mainly because of my interest in the area. So some of the things that I'm doing, I get asked on a daily basis to evaluate how much stool patients have. Kids are constipated. Everybody knows this. And we get x-rays. I don't know if it's the same here in India, but they love to do x-rays for this. And so I tried to develop a artificial intelligence to tell how much stool was on a abdominal x-ray. And this is an impossible thing for humans to do. And so I thought maybe an AI could do it. It turns out maybe not. So I spent hours and days and weeks highlighting poop on x-rays, thousands and thousands of x-rays. This is an example of a 3D slicer program that we developed to do that. Fed it all into a computer and got poor results. A little bit better results here. So I do a lot of abuse imaging. In pediatrics, posterior rib fractures are a major finding of abuse. And so I wondered if AI could find these better than humans, because we're not very good at it. We're pretty good when we're looking for it, but we forget to look at the ribs frequently on chest x-rays. And here are some examples of human and then AI detected fractures after a long time of training and many, many hours spent on thousands and thousands of fractures. And the AI does pretty well. We can get up to a 90% sensitivity depending on exactly the setting. Here's kind of an interesting example here where the blue is a human that outlined a fracture and the yellow is a AI that outlined a fracture. And I think the AI actually did find another fracture that's healing that the human didn't find. So this is going to be a useful thing in the future, but it's not quite there yet. Those are projects that I've been working on. But what is AI in general? If you go out into the world and you're not in the hospital anymore, and you're talking to your friends or to your family about artificial intelligence, the general public, they probably think of these things. So computer-generated images, chat GPT, large language models like DeepSeek is a new one that's just come out recently. Things like Spotify and YouTube, where it uses AI to give you personalized recommendations for what you might like. Facial recognition, robots driving self-driving cars, and unfortunately, maybe some misinformation like this picture in the bottom that is completely fabricated. and was used in the presidential election, along with a lot of other misinformation that was generated by AI. But in medicine, we're not talking about these things. We're talking about other things. We're talking about how can AI help us with things like workflow optimization, general diagnostics using labs or imaging, prediction and prognostication, talking about social and exam findings, and image analysis, what we were just talking about a second ago, patient screening. And there's a million different things that AI is doing behind the scenes that you usually don't think about, like voice recognition is using more and more artificial intelligence. When I want to talk about this, the Gartner hype cycle, is anybody familiar with this? Have we seen this before? It's kind of an interesting thought experiment. So Gartner was an IT firm that developed this, and it's not a qualitative thing. It has to do with the introduction of any new technology into a market. And so at the beginning, you have a technology trigger, so artificial intelligence. And then everybody gets very excited, and there's this huge peak of inflated expectations, and everybody thinks it's going to change the world. It's going to be this magic new thing. And then people realize, oh, it's not as easy as that. And then they go through this trough of disillusionment, like this here. But then we start thinking about it, and we start using it, and we start coming up with ways to make it useful. And we go through a slope of enlightenment and finally hit a plateau of productivity. Maybe not quite as high as what we initially thought it was going to be, but yes, useful in the end. And I think this is a useful way of thinking about AI. AI is obviously a huge topic, so where exactly and what exactly you're talking about, you may be somewhere on this cycle. I think I'm on the downslope right now for artificial intelligence. I'm generally pretty down on AI, especially how it relates to radiology and medicine in general. I think we have inflated expectations, and people are starting to realize that. But we'll get there. So in 2021, this is now 4 years ago, this was published in European Radiology, Journal of European Radiology, and you can see here there's just hundreds and hundreds of papers on AI. Each one of these papers is evaluating a specific product, talking about image analysis in radiology. This is just radiology, nothing else. And obviously this was 4 years ago. So you can do a PubMed search just to see, it'll let you get a sense of how many papers are being published on any topics. And I pulled this out of PubMed about 3 weeks ago. And you can see AI had kind of this baseline, a few studies here and there, and then in the last few years, it's exploded. So this is something that everybody is really excited, and this is an easy way to get published papers because journals know that this is something people want to read about. So where are we using AI in our practice? So this is specifically pulling from the University of Washington, which is an associate adult hospital that's near where I work. This is our work list. It's using Epic, and you can see that we have an AI doc, AI running in the background. It analyzed the images, and it flags some as important. It said specifically, hey, this patient has a fracture. And sure enough, the patient has probably a chronic fracture. So it will do the same thing for pulmonary embolism, or at least what it thinks is a pulmonary embolism. Now, it won't diagnose it. It won't tell you where it is. It won't put arrows on anything, but it'll say, hey, I think there's something wrong on this patient. And then you have to go look for it. But it will put that patient at the top of the list. Something that's more interesting for pediatric radiology is this. This is an MR image, thank you, on the left, using a specific sequence that was developed by a team that then uses an AI algorithm to turn that MR sequence into the equivalent of a CT sequence. So this is called Bone MRI. It's another company out there that will sell you their product. But I think to avoid radiation and double examinations, this could have a lot of potential. Now, it's not FDA approved in children yet, but it's getting there. This is something that is FDA approved in adults, not in children. It's called ClearRead, and we use this at the University of Washington as well. And you can see it did a really good job with this little tiny nodule right here in a patient with lung cancer. Hard to see, hard to pick up. You might just fly by this, but when you apply the AI and it goes through and removes all the vessels and airways, it really pops that little nodule out. So that's something that's very useful. This is the only AI-approved image analysis application in pediatrics. It's called Gleamer. This is an example of a subtle fracture that this AI found. So good job, Gleamer. In this case, I will say we've evaluated it, are in the process of evaluating it right now, and it's not always this good. So we probably are not going to be using it in a clinical setting, at least at this point. So those are some examples of AI in radiology. What are some of the problems? Well, for pediatrics, data. So in general, for both adults and pediatrics, there's just not enough data. So we need more. We need clean data that has been analyzed and gone over by humans so that we can train all these algorithms out there. Pediatric radiology is a small subsection of overall radiology, so there's just much less data for me, which is unfortunate. And that's why there's not very many approved applications that I can use. Model bias is a big one. So if you take all your data and it comes from one hospital or one country or one age or one gender, then that's not going to be very applicable to the larger patient population. And who owns all this data once you've got it? Who owns the algorithms? We don't really know. These are very gray areas. Monitoring this, doing QA, QI, all things that haven't been worked out yet. The legal implications, obviously, if I said that that kid didn't have a fracture and the AI did, and then it turns out that he did, am I going to get sued because I missed the fracture and the AI found it? These are all things that have yet to be determined. There's lots of problems. This is just a short list. What are some potential solutions? Well, there's lots of interesting ways, and there's very smart people working on these things. This is an example of something called data augmentation. This is something that we did for our rib fracture analysis. This is a kid with the initial X-ray on the left, and you can see there's a healing rib fracture, that little blob right here. That's the callus forming around the rib. And so that's one rib fracture. And we can feed that into the system and say, hey, learn what the fractures look like. But what we did instead was we said, hey, let's take that image and let's add some noise to it. Let's blur it, let's mirror it, let's do all these things that actually make it so that we have one rib fracture and we turn it into 10 rib fractures that all look a little bit different. And then we feed all 10 of those into the system. That allows us to get more information into the algorithm to train it on, and can be a really useful thing. Synthetic data, what if we just made up fractures? This is something where we took this image here, this is the same X-ray, and these are rib areas that did not have fractures. And then we said, let's create an AI that actually generates a fracture on each of these ribs and then feed it into the other AI. So this is like artificial intelligence that's building on artificial intelligence. Obviously, there's lots of problems in this kind of area, but it is something that you can do and we have done, and does give some value. Radiomics is especially useful in oncoradiology. This is interesting because it uses feature extraction for things that humans may not be able to see, at least able to see easily. Things like the shape and border features or texture of a image, or you can see over here in the gradient kurtosis of the lesion in this patient's chest tumor. Obviously, we're not thinking about the kurtosis of pixel values when we're visually analyzing something, but it's something a computer can very easily do. And so this is additional ways to add information into a system. Curated data sets do exist. They are available. There's lots of them that are free. Here's 5 that you can just go and download right now, the DICOM data, and say, okay, here's a bunch of tumors in the brain. I'm going to take all this data and make my own AI out of it. So I think things like this are very important. Making this freely available will help with a lot of these problems. Competitions. Here's 3 different competitions that are being run on an annual basis that you can go and join or have your IT departments join. RSNA AI challenges might be one of the biggest ones. People might be familiar with that. That's the Radiologic Society of North America. And they give data out and say, okay, people, go make what you can make out of this data. And similar to the Grand Challenge and openchallenges.io. So regarding some of the ethical issues, people are working on solutions to this too. So our hospital in Seattle Children's has developed a neutral oversight board, an IRB, and they call it the AIRB because they're trying to be clever, to evaluate each new use of AI in the hospital and make sure that it's done in an ethical and equitable way. It's a hard thing to do, but there are some guidelines from people, national, international societies like this one here, that can give you an outline of how to go about doing this. Other areas you can see here, transparency of data collection, data sharing agreements, informed consent. All right, so the future. So I think in the short term, these apps are going to continue to be developed. People are going to make these. I'm going to make an app that detects rib fractures. Somebody else is going to make an app that detects something else. Somebody else is going to make a better one of these. And you're just going to be able to plug them into your system, into your hospital EMR, into your radiology PACS system, like I'm showing right here. And these are just going to accumulate. And in 10 years, there's going to be thousands of these things out there, if there's not thousands already. They're going to get better and more useful. So I think that is definitely going to be happening. These are short-term things that you can see are going to happen right now. In the medium-term future, 10 years, 20 years from now, I think we're going to see things like this. So here's an example of two exams or two papers that were published a few years ago now. The paper on the left took hand radiographs that were done for bone age analysis. A lot of you may be familiar with these. And they actually said, we're going to feed all these into the system and let the system determine whether the hand belongs to a male or a female. And they found that AI could determine with 90% accuracy whether the child was a male or a female. That's not something humans can do. We still don't know how the computer does it. You can see here it's grouping them into male and female, and we gave they tried to determine where the computer was looking, but we really don't know how that is happening. But it's going to be very important for us to figure out how that works. Opening the black box that is an AI, there's lots of people out there that are trying to break down these algorithms to give us insight into how these things are working. Similarly, there's retinal photographs that you can take, and they did the same thing, trying to determine male or female with pretty good success. And that's not something that human ophthalmologists are able to do. The far future. So I'll start with a question, and we're almost done here, but let's say you give medical trainees 10 x-rays. You give them an hour lecture, and you give them some pizza. You set them down and you say, here's what pneumonia looks like on an x-ray. After that hour of training, looking at those 10 x-rays, they probably have about a 90% accuracy. They can take a new x-ray and say, okay, here's pneumonia. You give an AI algorithm tens of thousands of x-rays. You give it days or weeks of processing times. You give it tons of electricity, and then maybe you'll get a 90% accuracy. There's something different about a human and an AI in the way that we learn, in the way that we train. So here is a radiologist, and obviously you can tell that this is a computer-generated radiologist because they have a stethoscope. There's actually lots of good ways that you can tell that this is an AI-generated image, but I'll leave that to you to look at. This is not an AI-generated image. This is my son. I have family that lives in Australia, and we visited over the holidays, and we watched the International Australia versus India Test match. And so my son was inspired, and he was learning to play cricket with his cousin who's batting. And look at that impeccable form. That's probably the third time he's ever thrown a cricket ball. So let's say my son injures himself playing cricket. I know it's a safe game, but let's say. He needs an x-ray. He goes to the x-ray room. He gets an x-ray. The image is transferred to our radiologist, and she's looking at it. Well, that radiologist who's looking at the x-ray has to take into account, okay, the patient is differently positioned. Maybe he's rotated a little to the left. Maybe he's rotated a little to the right. There's other things that positioning can go in. She has to think about the fact that my son is a human who's made up of a bunch of different systems. He's got respiratory system, the musculoskeletal system, GI, cardiac. These all come together to make him the whole person that he is on a physical basis. Each one of these systems is prone to different pathologies. Here's a group of pathologies that might affect these systems, and obviously these are very complex, and I've just drawn arrows to kind of indicate, but they're going to be interplays here that are extremely complex. My son also lives in the world in a community where there might be pathologies present specific to that area the radiologist will be thinking about. He lives in a world where maybe there's a pandemic happening. I don't know that that could ever happen, but she has to use her knowledge of artifacts on the image. Is there something in the way? Is there something else? She has to weigh all of these different things in her mind, and she does it automatically, not thinking about these things on a specific basis. She takes into account her knowledge of physics, of teenagers in general, what kind of things happen to them, of normal growth and development, and she does it all within her lived experience. This is just a very simple thing that we do every time we look at an X-ray. In fact, every time you look at anything, this is going on in the background in your head, and you do it almost instantly. And this is something called artificial general intelligence, or in our case, general intelligence. And we are nowhere near this, but this is what we need to develop in order to train a radiologist, an artificial radiologist, to look at an X-ray. I don't anticipate that we're going to be here for at least another 50 years, if not longer, if not ever. I could be wrong, but I'm very optimistic about the job that I have. It's not going to be replaced by a computer anytime soon. So I think once you've gone through all that trouble, coding and artificial intelligence, the actual much easier thing to do is just train radiologists. So in the end, I think that's where we're going to be. We're going to have adjuncts, and they're going to help us, but they're never going to replace us. And that goes not just for radiology, that's any clinical field. We will not be replaced by machines anytime in the near future, which is good for us. So thank you very much. And I want to say thanks to everybody here, Dr. Akshay Baheti in particular, for inviting me. And here's my references. So thank you.
So much. That was an interesting talk, and there's a lot to think about. I think we'll take the questions now because you need to run away for another thing. So if we have some quick, intelligent question, naturally intelligent.
So I don't know if it's intelligent or not, but you mentioned accuracy. So do you have the same issue with hallucinations in radiology as other people have encountered in whispers transcription, all the other AI-driven?
Yeah, that's a very good question. I do worry about that with some specific applications. So hallucinations are when the machine kind of makes up a new image or a new word or something. In radiology, I specifically worry about it in the setting of voice recognition, where you are dictating a report, and some of the newer dictation systems out there use artificial intelligence to take what you say and make it into a standardized report. And so I worry that when it takes our words and standardizes them and rewrites them into a report, that it will have hallucination issues. And when you say, oh, there's no tumor here, it will say, oh, well, maybe there actually is a tumor here. We haven't had enough time with them yet to know how big a problem it's going to be, but it's certainly something that we're aware of.
If I can ask a quick question to you. One of the things that we face in our day-to-day practice is, evaluation of disease extent and response evaluations.
Yeah.
So where are we in terms of application of AI in assessment of disease extent and follow-up imaging?
For radiation oncology in particular, I know that there's lots of different companies that are working on this. Brain tumors in particular are being looked at because it seems like a relatively contained area that we could focus on. They've had some success, and there are some AI applications. Microsoft has developed one with a private spin-off company, I believe, that will try and segment out an exact area of tumor involvement within a brain tumor. But I believe, we don't use it in pediatrics, so I don't have personal experience with it, but I believe it's not accurate enough. It does not do a good enough job to be useful for us clinically yet.
One of them is the extent of pulmonary nodules, 5 nodules, 6 nodules, you give chemotherapy and then you're doing a response assessment. So if you have an AI mechanism by which you can actually do that count and compare, it would be easier for our radiologists to spend more time on other things rather than counting metastasis numbers.
Yes, and I think radiologists would love to get away from counting metastasis. There are some applications that will identify, like we saw here, the nodules. As far as I know, they don't count them yet, but that's probably coming very soon. Yeah.
Thank you so much.
Thank you.
It was really nice talk. So we'll move on to the next talk. Which way do we go, Jeffrey, first? Okay. So I'll invite my colleague, Dr. Jifmi Jose Manjli, and she'll be speaking about unknown to known in pediatric normal tissue tolerance, insights from the PENTEC study. Over to you, Jifmi.
Thank you. So shifting gears from diagnostics to therapeutics and also from the tallest speaker in the EBM to the shortest speaker. So, okay. So are we going to presentation mode? So before I delve into the topic, I would like to acknowledge this a huge group of people who have put together, put so much effort over a decade, maybe 2 decades, and come up with such, data for us so that we can understand, we can implement this on our patients and also further develop on this. So can we go to the next slide? Yeah. So my flow would be that we look at the premise, compare it with Quantec, see what Pentec is, look at the challenges, the aims, the work and task forces, and come to some dose volume parameters that they have given to us. So to begin with, we've been discussing about all this, that around 36 million of children get diagnosed, most of them concentrated in Asia and Africa. And despite having around 80% survival rates, we get to know that around 90% of them have some kind of chronic health condition that they suffer from. And radiation in particular causes a twofold increase of mortality. So this is a picture of Dr. Giulio D'Angio, who were the pioneers to, develop this thought amongst the radiation oncology community and maybe the whole pediatric oncology. He was also a pioneer in developing the NWTS studies. And this is what sowed the seeds in many of the youngsters then, now quite old. So Dr. Sandy Constant is spearheading the Pentec, and he took up this from there. So this Quantec is something that is described in the adults. So it is the quantitative analysis of normal tissue effects in the clinic with radiotherapy. So why can't this be just applied in children? So you can always go with the art of antiquity, thinking that children are the miniature versions of adults. But radiobiologically, it may be true in general, but radiobiologically, this doesn't hold any value, because we must understand that in children, we have a complicated relationship between the spatiotemporal distribution of dose and the toxicity profile that we see. That is essentially because of the mosaic of tissues that children have. And by mosaic of tissues, we mean that you have tissues developing at different rates and at different temporal sequences in a particular organ itself. So a little about the functional reserve between adults and pediatrics. So by functional reserve, I mean the minimum amount of functional units in a particular organ, so that despite an insult, it can continue functioning normally. So in adults, what we understand is that you have inherent structures, and each organ is divided into stem cell, progenitor versus terminally differentiated, and it depends on the volume and age of the person. But when you come to pediatrics, despite these factors, you have additional factors as well. You have contribution and extent of the proliferating compartment very significant. There is a lot of developmental dynamics, as I mentioned before, that it varies between organs. You need to look at the regenerative potential, which again may differ between different organs, and senescence, of course. So this varies between organs, between the same patient and also between different patients or cases. So so what we need to understand is that while the cellular and subcellular impact of radiation in adults and pediatrics are the same, the tissue remodeling and the functional consequences of radiation injury differ markedly. So that's how gave the genesis of Pentec. It is definitely an offspring of Quantec and a sibling of High-Tech. To the non-radiation oncologist, it is again an evaluation of hypofractionated radiotherapy in the clinic. So it was developed with a resolute recognition that all our survivors have a huge impact due to the normal tissue damage and subsequent malignancies that develop because of radiation. So they try to have a systematic overview and a meta-analysis in cases where possible about the dose-effect relationship, and also on adverse effects in survivors. So they developed ways in which they could synthesize normal tissue constraints and also assess the impact of chemotherapy and other risk factors on these. So I could just enumerate the aims, that is again, quantitative evidence-based dose volume parameters, estimate the quantitative effect of each more relevant risk factor, describe the specific physics and dosimetric issues behind them, propose dose volume outcome reporting standards, and also define future research priorities. So enumerating them and talking about them is very easy, but coming to work is really difficult. So all the investigators involved in this did realize how challenging this could be, and they did acknowledge before they embarked on this journey only. So the initial reports do elucidate this. It's not that after the reports came out that they understood that they are the challenges. They knew it beforehand. So many of the challenges, a few of them I would just delve on, is that the long-term adverse effects, that the data that we sought for is over maybe 3 or 4 decades. So during that time, the therapy, the complete therapy, including chemotherapy, surgery, and radiation, would have changed quite a bit. There would be the techniques that you look into, that you use now is IMRT, but then would be much more primitive, relatively primitive, that is the 2D-RT and 3D CRT. So that would have changed. You do not have much of CT information. So if you have to have, this would be a general understanding that if you have to have a dose volume relationship, then you need to have volume data. So that comes from CT. But we must understand that the decades from which the data is analyzed. So this is usually from the late 90s. The data that is analyzed in Pentec is from the late 90s to the late 2010s. So the reports that were published in this 2 decades. So the reports that are published here were actually of patients treated maybe from the 1960s onwards. So they are definitely using primitive techniques. So how much data we could derive from them when they actually have not been based on volume related reporting. You actually do not have uniform standards in reporting toxicity. Even there is no consensus on how to report the dose volume parameters. So you have multiple issues, you have confounding factors. There is a problem with dichotomizing these outcomes. So many of these outcomes, we know that they are continuous, like growth retardation or mental retardation. You know that this is not like yes or no, it is a continuum. But because we need to analyze them, they have to be dichotomized so that you have a lot of data, loss of data. So then also the late effects may not be specific to the treatment itself. And you have behavioral risk factors. You need to know about retreatment, which again, you do not have much data on. There is a lot of gap in having information about substructures. Like if you're looking at cardiac toxicity, each toxicity is related to the dose given to that particular substructure. So that is, I mean, they initially only knew that they are not going to get much data on that. And also according to the timing of exposure in a particular patient at a particular age, it's going to have an impact on how much toxicity they're going to have. And also the relevance of low dose versus peak or mean doses on subsequent malignancies in children. So despite knowing all this, why do we think that this is still relevant? It's because despite the primitive RT techniques, we know that much of the patients are in Asia and Africa, and many of them are still treated with these primitive techniques. So knowing that is very important. And also, again, like how Dr. Banavali elucidated too, we need to know the past so that we can predict the future. And we could have some reasonable estimates to what we have to look for and also take care when we are planning. So we had a huge group of people working towards this. They were divided into task forces. So you had a bunch of physicians, you had, including radiation and pediatric oncologists and other specialists. We had medical physicists. We have mathematical modelers to do the modeling, and also epidemiologists to know how the distribution is. So they were put into 18 organ-specific task forces. They looked into the clinical question, divided into PICO, that's the acronym standing for Patient Intervention, Comparator, and Outcome. They looked into the literature review. They set inclusion and exclusion criteria for the studies. They abstracted the data, looked at the risk of bias of each study, and also did data analysis, as I specified before. Most of them were systematic overviews, and where possible, they did a meta-analysis. So they decided to develop 7 introductory reviews, around 18 to 19 organ-specific reviews, and have visionary reviews as well, so that this could have an impact on the future of radiation oncology. So coming to a bit of the organs, I wouldn't be discussing everything because of the paucity of time. So I'll look into maybe 4 or 5 organs, and this would be described in the order of which they were published. So the initial reports included that for the breast. So what was analyzed is that they looked at the dose, the partial or the full dose exposure, RT exposure that happened to the mammary annulage. That's another word for breast bud. So it's a very small structure. In a small individual, it would be just like 3 to 4 millimeters deep to the breast, the nipple. So they looked into the doses, and what they found, they looked into the endpoints that they looked at was hypoplasia and lactation. So in hypoplasia, they looked at patient perceived asymmetry as well as volume difference. So I would just go to what would be significantly impacting them would be the maybe a dose more than 1.25 looked at more than 20% of asymmetry. And also lactation failure, which meant even one, I mean, for the ease of reporting, you can define it in multiple ways. I mean, it is not a single definition, but what they looked at was just one single attempt of failed lactation. So that's what they looked into. And they found that 27 to 46 gray exposure led to around 40% of failed, unsuccessful breastfeeding. So it's not only hypoplasia and lactation failure. They did acknowledge it causes a lot of other impacts like social discomfort and psychological distress. But obviously they couldn't evaluate this because that was not reported at all. So coming to cardiac, this was realized as the most common and severe effect, late toxicity system. And when you look at the radiation dose, so a dose, they looked at the absolute risk after 30 years of radiation with a dose of 20 gray. So the percentage, the absolute risk for any cardiac disease was about 4%, as you see here. And the most common was CAD, coronary artery disease. So they also tried to find an equivalence of 100 milligram per meter square of anthracycline with radiation and found that to be around 10.5 gray. So this was quite complicated because we know there is a confounding factor of anthracyclines. Though they took it into consideration, there's still, you do not know whether it is additive or synergistic or so. So we did speak a lot yesterday about endothelial damage and how this relates. This goes on to fibrosis and late cardiac effects. So that's about it. And moving on to lung, they looked at the clinical and self-reported pulmonary outcomes after radiation and found that when the mean lung dose is about less than 14 gray and V20, that is the volume of lung receiving 20 gray is less than 30%, the grade 2 and grade 3 pneumonitis was controllable. However, the toxicities, the pulmonary toxicities was slightly higher when you looked at whole lung radiation. Also, bleomycin and other chemotherapeutics did not have an impact when partial lung irradiation was done, but it did have when there was whole lung irradiation. Coming to kidney, they looked at whole abdominal irradiation and also TBI, also partial kidney. So what they saw was, as you can, I've enumerated the endpoint and the percentage of toxicity. So the toxicity was slightly higher when it was TBI, while it was around grade 2 was 4% and grade 3 was 1%. Here it was 8 and 3%. So this is probably because of the higher doses of chemotherapy and the intensity of treatment you need to. So TBI is usually given as a conditioning regimen. So probably it is because of that. And when you look at partial kidney, when the dose was kept around 5 to 10 gray with or without carboplatin, the expected toxicities was less than 5%. So overall, what they suggested is the NTCP could be kept around 5% when the less than around 5% when the doses to the kidney was kept at 8.5, 10.2, and 14.5. So this is to an extent similar to what we practice daily in clinic, is that we try to keep the mean dose to the kidney around 10 to 12 gray. So this wasn't a surprise. And also about the vertebra. So there is a lot of discussion about vertebra because it does impact the height of the patient and also causes scoliosis or not. So what they came across is that around 15 gray in less than 2 years did lead to scoliosis, a significant proportion of scoliosis. And they did suggest that we need to have, I mean, the variation between the parts of the vertebra should be less than 10 gray. That is essentially to avoid asymmetrical growth. So coming to CNS, the subsequent IQ less than 85% was seen in 100, I mean, 100, when 100% of the brain was treated with 18 gray, and that is what we usually use in cranial RT. And the other important point was they tried to find the equivalence between methotrexate and RT, so that was found to be around 5.9 gray. Coming to spinal cord, the toxicities were seen around 50 gray. Again, that is what we use. But when we go to smaller segments, as in smaller volumes, 0.03 cc, we can go to higher doses of 54 gray. And going to the reproductive system, a significant ovarian failure was seen at 2 gray. And they did correlate it with the cumulative effective dose of cyclophosphamide. And they did see that it is much more, it happens at a higher dose at smaller age and yes, lesser dose at 20 years. So uterus was thought to have stricture, I mean, they were impacted when the dose was more than 12 gray. Testis, the scatter dose of less than 1 gray led to a transient oligospermia, but when it was more than 1 gray, there was 90% of permanent oligospermia. So I'll just quickly go through the systems. That is, there was significant endocrine impact when the dose was more than 21 gray to the HPA, that is hypopituitary axis, hypothalamic pituitary axis. And so SMN, I'll just take 10 seconds on this. So the majority of them were meningiomas and CNS malignant lesions. So they occurred at around 20 gray, from 20 gray onwards. Obviously, the percentage increased when 50 gray was given to the brain. And this happened at a latent period of 10 years. And it was about 4 to 10 gray, 10% with 50 gray, and 2 to 5% in 20 gray. Meningiomas happened much more commonly, more than 25% when you gave even 24 gray. So that happened at 20 years. Sarcomas are much more less common, and as well as lung cancers. So coming to the conclusion that functional consequences in children is significantly high. They are affected by total and fractional doses of radiation, dose rate, overall treatment time. They are affected by cytotoxic and molecular targeted agents as well. As Pentec has come about with all these articles, they do suggest that it is not for absolute cutoffs, but to describe that this is a continuing, I mean, it's a continuum of effects, and we need to look at it that way. So we do have to have this information so that we can have a shared discussion with the patient and explain to them that this is the risks that you have if we have to go with radiation. So that's about it. Thank you. This is the website that you can go ahead and look at all the articles of Pentec and their future programs. With that, I do end, and I also like to invite all of you who's interested. I mean, I know majority of the crowd is non-radiation oncologists. For our annual pediatric masterclass that happens around September, we would be putting out the flyer soon. Thank you.
Shaping pediatric radiotherapy for the future. Over to you, Neha.
Thank you all for being here. Radiation oncology, or planning radiotherapy for any patient, be it adult or a child, is a complex procedure. It requires intense research and acknowledgement of drawbacks versus benefit of evaluating a treatment plan before it is executed or implemented on a patient. There have been several breakthroughs in the last 2 decades which have evolved from experimental to something which has been incorporated in daily practice. Along with that, there are certain breakthroughs which are in the pipeline. So the vendors, the radiation oncologists, physicists, and the engineers have been really working on technological aspects on improving what we call is conformal radiotherapy. So when I say conformal radiotherapy for non-radiation oncologists here, we conform our radiotherapy volumes to the complex tumor volume. And when we do this, we try and avoid or spare radiation, unnecessary radiation, inadvertent radiation dose to the adjacent normal tissue. And when I say adjacent, it's not just close, but when the patient of interest is a child, the integral dose is also very important, which is the low dose which is received by the entire body, not just the adjacent critical normal structures. IMRT and volumetric modulated arc therapy, these are the standard of care for at least a decade. This has been in practice for almost, even in peripheral centers which have radiotherapy. So very few patients are treated with conventional way of delivering radiotherapy. If you look at the example of Hodgkin's lymphoma, how the radiotherapy portals evolved from mantle, which was an extensive large volume, to something which is called as involved site radiotherapy, something which was treated with conventional portals to something which is treated with much more conformal way of delivering treatment. And you will see that in this study, the AHOD study, 75% of patients were either treated with IMRT, VMAT, or proton beam therapy. When I talk about brachytherapy, brachytherapy remains the most conformal way of treating a child, though this form of delivering radiation is much more uncommon as compared to the external beam radiotherapy, but it still remains the most conformal way of delivering. In this paper from TMH, Dr. Laskar highlighted the evolution of brachytherapy in the last 3 decades, not just improving the oncological outcomes, reducing the toxicity, but what it highlighted was brachytherapy has a learning curve. And as in the recent years, more and more complex cases, more and more younger children, more and more cases with plastic reconstruction were considered. And of more than 100 patients that were in this review, there was just 1 case of radiation-induced second malignancy. When I talk about stereotactic body radiotherapy, it's the standard of care in adults, but its incorporation in pediatrics, it's still limited. If I look at the Ewing's, Euro-Ewing's R3 data, it says that in oligometastatic and metastatic Ewing sarcoma, if a metastatic site is treated with local therapy, there is an improvement in outcome. That means the primary site as well as the metastatic site have to be incorporated in your local treatment protocol. And if you look at few current radiotherapy treatment trials, so metastatic rhabdomyosarcoma, Ewing sarcoma, they all incorporate radiotherapy, especially in the form of stereotactic body radiotherapy. This very interesting study, Lightsaber, I would want all the residents to go through this data. It evaluates the role of SBRT, and the most common histologies were Ewing sarcoma, osteosarcoma, neuroblastoma, and rhabdomyosarcoma, and other non-round cell sarcomas. And the most common site was spinal, paravertebral, lung, apart from the other sites. It said that SBRT led to a local control, a 2-year local control of 74%. But it also emphasized that the dose of SBRT that you give has an impact on the local control. It is not that you treat the primary tumor with full dose and then to the metastatic site you give 8-in-1, 12-in-2, 20-in-5. No, it cannot be like that. That dose has an impact on local control. And if you look at the extent of impact, every 10 gray increase in the BED dose has a 5% increase in local control. This National Pediatric Cancer Foundation involved radiation oncologists who were involved in academic institution in US. They established answers to critical questions with respect to SBRT. And it says that if patient is planned for treatment with curative intent, the primary as well as metastatic sites should be treated with higher doses of radiotherapy.
Up to 8 sites can be treated. Of course, this is case by case. And regarding the sequencing of this kind of radiotherapy, you can do it for logistic reasons. You can do it with the radiation for the primary site or at the end of completion of systemic therapy. Dr. Jifmi did mention about the Pentec dose constraint for the vertebra. You have to understand that whenever conformal radiotherapy is used, of course, doses to the adjacent organs are reduced, but this happens at the cost of a gradient of dose along the bones, especially the vertebra. And this gradient or dose heterogeneity in the vertebral body leads to risk of kyphoscoliosis and other problems like compression fractures and all that. So this paper, which comes from the SIOP Europe, explains us the location of growth centers, how to contour vertebra and other bones, and how to avoid this kind of gradient, especially in children who have not completed their growth spurt. Cardiac sparing whole lung radiotherapy, its name is a giveaway. Whenever we give lung baths to our patients with Wilms tumor, we have to remember that the third drug which was added to the two-drug backbone for stage 3 and 4 Wilms tumor is adriamycin. And any dose of radiotherapy that you give to the heart when a child has already received adriamycin is going to increase the risk of late cardiac morbidity. And with the better accessibility and availability of IMRT and BMAT, most of these patients are treated with cardiac sparing radiotherapy. If you look at the data, the data is not that strong enough. Not many cases have been treated, but in institutes like St. Jude's, which practice proton beam therapy for most of their patients, they have these protocols wherein not just the heart, but even the breast can be spared of the radiotherapy dose when it is planned for lung bath. Deep inspiration breath hold is an extremely important way of delivering radiotherapy to mediastinum, and it is commonly used in adults, especially for left-sided breast cancer. I think almost all patients, even in our institute, are treated with deep inspiration breath hold. Now, it is always known that, it is difficult for children to understand the breath hold technique, but I came across multiple papers wherein children who were even 5 years of age were able to understand the technique of breath hold, were able to maintain that breath hold during the radiotherapy. And for the non-radiation oncologist here, of course, when in the inspiration mode, the chest cavity increases, the lung volume expands, the diaphragm is pulled down, the heart becomes more elongated, fusiform, and comes a little away from the radiotherapy portal. So more and more utilization of DIBH will have a significant impact on cardiac toxicity for especially for hematolymphoid malignancies where mediastinum is frequently treated. For TBI, Dr. Jifmi did mention doses on kidney when TBI is delivered. So in most centers, TBI is offered with a very conventional way of delivering radiotherapy with lung blocks. But with VMAT, the TLI protocols that are existing can significantly reduce the doses to the kidney and the lungs. Dr. Laskar in his talk yesterday did emphasize on how protons have been important in optimizing outcomes in children as well as reducing the late toxicity, especially with respect to second malignant neoplasias. But proton therapy comes with its own set of uncertainties. It is a complex technology, a very labor-intensive procedure. But there are continuous advancements that are being made in this technology, and I will talk about some of these advancements. The most important aspect was shift from the passive scattering to pencil beam scanning, which kind of really helped in reducing a lot of uncertainties in the delivery. And whenever we talk about proton therapy, it becomes all the more relevant in children, because in children, you would not want to have that kind of uncertainties with proton beam therapy. I have given an example of an IMPT, which is intensity modulated proton beam therapy for CSI. So what it does, you can use different techniques, but in this publication, there was a special technique which was used, which was called as the split target gradient technique. What it ensured was a very soft transition between fields, and a matchless or a junction-free CSI radiotherapy could be offered to these children. The other uncertainty or the other concern when it comes to pediatrics is the increase in the LET at the point of Bragg peak, which results in undesired heterogeneity, especially across the proton field. So incorporating this dose-weighted LET is extremely important when you plan. So there are different optimization models that are available, and the current model that most of us practice right now is the robust optimization model, which kind of helps us to modulate or play with this high-density LET region. It helps us to manage it properly, sometimes even put that LET region in the tumor and not in a critical organ like brainstem, where it can lead to significant morbidity. You can see that in this picture also, that if you incorporate a biological effect-based objectives to robust optimization, it can really help in the face of managing these range uncertainties, the setup uncertainties, and the tissue heterogeneity for proton plans. Lateral penumbra becomes especially important in children for superficial tumors like orbital tumors. The advancements are made in terms of, adding aperture collimator to make the fields more sharper. Another concern that was there with passive scattering was that of neutron contamination. And it is seen that with pencil beam scattering, the neutron contamination, which is a high LET radiation, the neutron contamination is being reduced to 2 to 3 times of what it was when passive scattering was used. Dual CT, the dual energy CT scan, is also something which helps us in differentiating different types of tissues. So once we know what this tissue is, if you use 2 different energy CT, every voxel can be assigned a CT density in a more appropriate way. And once that CT density is assigned, we can know better how the proton beam is going to interact with that tissue. And it really helps us to overcome the range uncertainties or the tissue heterogeneity that we face in certain sites, as well as whenever there is an implant. So whenever we talk about these advancements in proton beam therapy, what is important is to understand all of this is done to make our volumes more conformal, to reduce the normal tissue that is around the tumor, to reduce the doses to those organs, as well as to reduce the integral dose, and in turn reduce the chances of second malignant neoplasia, which happens to be the most supreme goal in planning radiotherapy for children. Now, there are these 2 buzzwords when we talk about radiotherapy, the flash radiotherapy and the spatially fractionated radiotherapy. These are all, technologies which are in their preclinical or early clinical phase. They are not implemented in pediatrics right now. Flash is when you give very high dose of radiotherapy. Spatially fractionated radiotherapy, when you convert the beam into microbeams, you have a very inherent dose inhomogeneity in the tumor, which in some way improves the local control, and some unknown mechanism, it also reduces the toxicity. So these, when they come to pediatric radiation oncology, it's going to take some time. Another thing which is very high energy electrons, which are beneficial for deeper tissues, again, something which is an emerging technology. Carbon ion and multi-ion, we did discuss. Dr. Laskar has recently got approval for a 1,500 crore multi-ion machine. So all these are ways and means to reduce late toxicity. Carbon ion and multi-ion are technically very complex, and they will have their issues about accessibility, affordability, and generating evidence for use in pediatrics. When I talk about image-guided radiotherapy is when we use CT scan just before the treatment to ensure precise delivery of radiotherapy to children. Of course, these are ionizing radiation, and there is a concern with ionizing radiation to children. So there are certain protocols which are coming up, which is called a soft IGRT, limited arc IGRT, which reduces the dose to the adjacent normal tissue. the MR-Linac is also coming up for certain tumors, especially the GI malignancy involving, so the liver, essentially for liver tumors and prostate and all, it will find an important place in planning radiotherapy for children as well. In this, the other aspect of using image-guided radiotherapy is also to reduce the safety margin. As we reduce the safety margin, we can reduce the amount of radiation given to the adjacent normal tissue. And it is in this very interesting paper, which was for Frank Carty in Wilms tumor, it was seen that the safety margin or the CTV to PTV margin could be as less as 2 millimeter. Combination with immunotherapy and radiotherapy, apparently it's still evolving in children. There are certain sites where it is kind of more or less established, for example, for neuroblastoma and for hematological malignancies. But for other tumors like sarcomas and nasopharynx, the trials are still, the results are awaited. There are, if you go through a PubMed search, there are tons of advancements in radiation oncology, all of which are a seminar in itself, and they are going to have an impact on the future of radiation oncology. I'll not will not discuss them. I would like to thank Dr. Laskar and Jifmi in helping me prepare this presentation. And again, let you know of Trellises, which is our annual radiotherapy masterclass. I would urge that the radiation oncologist sitting here and the non-radiation oncologist should urge their radiation oncologist and their team to come and participate in this program. How we learn from each other, all radiation oncologists, is going to shape the future of pediatric radiation oncology. Thank you very much.
Thanks, Nihal and Jifmi. Excellent talks. This is a question that either of you can answer. So we heard so much about pharmacogenomics yesterday in the development of late effects. So can you tell us something more about radiogenomics and whether it's actually in use or whether it is of utility?
I actually wouldn't have an answer because I think I rarely read about that, and it's actually in very, very primitive stage. Yeah, primitive stage. And we have, in fact, not started using it at all. So we need to, especially in children, when you have good dose volume parameters is when you can have those kinds of applications. So there's a bit of dose painting and all that stuff. So all this is impacted with a lot of tumor heterogeneity, and if not tumor, the normal tissue heterogeneity. So unless you have all that charted out and we have some kind of way in which we can know better. The other issue with this is that this can change over time. So today you may see it as tumor and it must be solid and partly cystic, but it can vary over the time. So if we have to have radiation put to these effects, then we need to have more data. So I won't be.
Dr. Banavali.
Yeah, this is again for any one of you. I wanted to ask it yesterday, but couldn't when Siddharth was talking. Again and again, you all stress when you give radiation, the local control rate is excellent. But sometimes, when you give local radiation, the local environment may become so bad that the tumor gets out and gets metastasized. So whether it's enough to, when you use radiation, and that's why maybe the outcomes of radiation versus surgery are much different. So can you say that if only the radiation gives local control rate is important, even the metastasis is as important in evaluating the efficacy of radiation?
So thank you, sir, for bringing out this important point. If you could make out yesterday when I was talking about protons, I did mention one thing, that when I compared the outcomes of proton publications in pediatrics, we find that most of them, and I did mention that repeatedly, the local controls are 80% and above. And even the metastatic rates are lesser. I showed you another study at the end, the group from Birmingham. They were using preoperative radiation therapy with protons in Ewing sarcomas, and their outcomes were much better. And the third thing that we find is that the patients who develop metastasis seem to be lesser in patients who were treated with protons. So the problem is that the volume of normal tissue that you end up radiating with protons is much lesser compared to the volume of normal tissue that gets radiated with X-rays. And that probably has an impact on the tumor environment, and probably in the long run also has an implication in terms of metastatic disease that you have. So that is, in a way, the systemic effect of local radiotherapy besides the local treatment.
So maybe it would be important to stress on the fact that proton not only improves local this thing, but decreases
Probably.
mets also, because that's an important information, you know.
Probably.
We also describe this phenomena as abscopal effect, and it is seen more
Scopal
with
is different.
protons and
Scopal is
Scopal is
slightly different.
Entirely.
You know, you would have an impact on that. We are combining with immunotherapy when you are seeing
You know, that's why I always say, like, abscopal effect ke liye you require only say 3,000 rats, you know, we are only killing the immune cell. But then you'll say that when you give the whole radiation, you're already killing the immune cell. But when you give the whole radiation, it causes a lot of local inflammation, infection, and brings in your Tregs and all the bad immune cells, you know. So abscopal is an absolutely different concept than this.
This is going to have a huge impact, I feel. When we treat with protons versus X-ray radiotherapy, the dose that we deliver to the primary tumor is the same in both. It is the same. The only thing that is changing is the low dose bath to the surrounding area. And in that, we've been asking two questions. Does the low dose bath impact development of second malignancies? And second thing is, does the low dose bath have an impact on development of metastasis or not? So there are two things that people are looking at.
This flash therapy and pencil beam, they say that it is a sort of a, like, as we talk of immunotherapy, systemic immunotherapy. Now there is a lot of talk about immunoradiotherapy, like how radiotherapy can be utilized or manipulated to act as an immune therapy as well. Can you just tell something about that?
If you really ask where does, radiotherapy is being combined in a way, we are using it in combination with immunotherapy in lung cancers, a lot of head and neck cancers we are using. It may not be as much as in pediatric tumors. We're using radiotherapy in combination with CAR T cell therapy. We are using radiotherapy is a very important tool as a bridging modality from the time you diagnose, plan, and from the time you do the CAR T cell. The bridging time is we use radiotherapy in various ways. And we have been able to cone down to a dose of about 30 to 35 gray in lymphomas as a bridging. So we are looking at these aspects where we can combine immuno with radiotherapy with a synergistic impact. You asked about flash therapy. Flash therapy is still not become clinical, but there is a lot of work being done. And the idea is that in flash, you're delivering way, way high dose of radiation in 1 second. And we truly don't understand what is the biological mechanism that is underlying the flash effect, where you kill the tumor and do least effect to the surrounding normal tissue. So we still don't know, but people say there are multiple mechanisms, and one of that is a rapid, consumption of all the oxygen that is there in that area in a very short time. That actually, in a way, protects the surrounding normal tissue, and the other effects actually destroy the tumor in a way. But we are not sure about how actually flash effect works. But flash, there's a lot of work being done in electron flash and also proton flash. So electron flash has been being done for some time. Proton flash is coming up, and maybe in the next few years, they would become clinical. There are some skin tumors that were treated with proton flash, and they've seen good responses and little toxicity. The other thing that Nehal mentioned about, you mentioned, madam, regarding pencil beam scanning. A lot of the proton data that we have now, the long-term follow-up proton data, was patients treated with the scattering beam technology that Nehal was mentioning. In scattering beam, what happens, there's a scattering foil, and the proton beam falls that, it scatters it. And because of the scattering effect, there's a lot of neutrons also that are produced from there. And these neutrons have a negative impact in terms of second malignancies. And now the modern machines do not have a scattering. They have a pencil beam of proton that comes and shapes and treats. So you are removing all those neutrons that were actually probably nullifying the benefit of protons. And that's why the Mass General Hospital data that we have, which did not show a huge difference between the second cancers between patients who were treated with protons versus photons. It was 5% and 6% between these two groups. It was not significant. But we need to remember that these were patients who were treated with the scattering technology. And now we have patients who are treating with the pencil beam technique. So probably in the next few years, like I showed yesterday, there is some data to show that there is a difference between IMRT, scattering proton technology, and pencil beam proton technology. So technology is making a difference. How big is the significant difference is to be seen in the future.
No more questions. So we will go ahead with the next session. I thank the chairs for chairing this session, Dr. Nehal and Dr. Laskar. So the next is our residents have taken equal effort to make posters and oral presentations. So we will go for the oral presentations, and I invite the first presentation by Dr. Shubham Kumar. So each one of you will have 8 minutes to talk. Shorter is okay, and then 2 minutes of questions.
I request the judges to step forward. Dr. Vikramjit Kanwar, Dr. Nehal Khanna, and Dr. Purvi Kadakia-Kutti. Please, if you can come forward. Thank you.
Start. So good morning, everyone. So my topic for today is it time to integrate dedicated pediatric cancer genetics unit in outpatient unit for detection of cancer predisposition syndrome in children. So this is our experience from TMH Mumbai. So we will go into a background. Around 10 years back, the proportion of cancers in children attributable to an underlying genetic syndrome or inherited suspectability was unclear. And pediatricians considered children with cancers for inherited cancer suspectability according to well-established clinical criteria only. So this was a landmark paper published by Zhang et al. So they showed a prevalence of CPS of around 8.5%. And so the new thing in this paper was that genetic testing can cover underlying cancer suspectability syndrome beyond family history. So we should understand the importance of knowledge of germline mutation. Why should we know it? So it helps us in better understanding of tumorigenesis, and it can direct patient care in the way of therapy modification, donor testing and selection for stem cell transplant, risk-reductive surgery, and pre-symptomatic benefit in the form of improved survival and minimize morbidity. And it can also guide us in genetic counseling. So the aim of setting a genetic clinic was to integrate genetic services into the routine care can have far-reaching consequences in identifying patients and family eligible for screening and early surveillance. Data from India is very few and limited to case reports and series. So we studied the impact of a dedicated PCGC in the outpatient department. So we started a pediatric CGC in around September 2021 with a certified genetic counselor. We defined a target population based on the CPS risk, based on the tumor characteristic or the patient characteristic, was screened, and if eligible, were tested for post-test counseling. Testing was done initially with a 26 gene Sophia gene panel, then 94 gene Illumina panel, and 84 gene extended Sophia panel was used later on. A post-test counseling was provided, whoever was required, in the form of extended family testing and surveillance whenever it was required. And at last, the pediatric CGC services for 2 years from October 2021 to hospital CGC services from 2018 to 2019 was compared. So this is the result. The pediatric CGC, we registered around 255 patients, out of which 218 were tested. And as compared to the hospital CGC, which was, we registered around only 58 patients and only 12 samples were tested before that. So this is the profile. So most of the patients were first primary, around 98%, 250 patients, and only 5 patients were with second malignancy. And the important thing to note is that only 27% of the patients gave a positive family history. And so around 64 out of 218 samples that were tested were positive for CPS, and the VUS reported was around 17.6%. So this is the profile. The CNS tumors were the largest that has been tested. It's around 24.5%, and it identified LFS, NF1 in varying proportion, NF2, TSC, CMMRD, and Lynch syndrome. Then next was soft tissue sarcoma, that 32%, retinoblastoma 33%. So various genetic syndromes were identified. So I conclude that there is a need to look for CPS beyond family history criteria. And there is need for integration of genetic services into the clinical care in childhood cancer. And the data on the prevalence and spectrum of CPS obtained can inform further future policies. So that's my presentation.
From the judges.
So that was defined on the tumor characteristic, that if the tumor is multifocal or any tumor in childhood that doesn't fit according to the age, any adult tumors in the childhood or some specific tumors such as ACC, choroid plexus carcinoma, or retinoblastoma. So it was identified from the McGill, but not based on that. The McGill score that we used for screening, but it wasn't based on completely on that. It was based on the clinician preference, if they can consider a positive family history or something.
So at one slide you mentioned hospital genetic versus pediatric. Yeah. So what was the criteria? I mean, in other words, I was trying to, was he overlapping? These were completely separate?
No, that's hospital CGC is 2018 and 2019. After that, we set
So this was the baseline prior to...
after that, we set it dedicated pediatric CGC. Before that, before 2021, there was
So it was
not
mixed adult and pediatric prior to that?
Prior to that, it was it wasn't dedicated. We just used to refer based on the.
Got it. So it is just basically representation as to how many you were testing prior to that?
Yes, sir.
It's roughly 50 per year.
Yes, 15 2 years, 58.
Can I ask one this thing? So you showed 27% had a family history,
Yes,
and
sir.
29% at CPS. So what was the percentage of family history who were showed CPS versus the other?
That data we have to analyze that. Don't have the exact.
Why I'm asking is, like, if you had most of the patients who had CPS from the family history, maybe that group requires testing rather than. So just to know that, it will be important.
We'll invite the next speaker, Kalashekar.
So we are talking a lot about including technologies. in improving cancer care. So this is one attempt in adding adding flow cytometry in the CNS leukemic detection to improve the diagnostic precision in pediatric acute lymphoblastic leukemia. So this study is funded by Lady Tata Memorial Trust Institutional Research Grant. So there is no conflict of interest to disclose. So background is CNS leukemia incidence by using cytomorphology. It is in children is up to 10%. So cytomorphologic assessment is subject to intra-observer variability, may result in underdetection of baseline CNS leukemia. So we actually don't know the true incidence of CNS leukemia, which may be actually higher. So also majority of CNS relapsed ALL patients, if you see upfront, they are actually CNS1 at primary diagnosis. So there is need for more sensitive techniques in CNS leukemia diagnosis. So this is one upcoming evaluation, which is including flow cytometry in the CNS leukemic detection. So preliminary data or preliminary research studies are showing that it is highly sensitive, and it is being proposed as an alternative approach for CNS leukemia detection. So this is a data from NOFO group. So they analyzed and show that it is able to predict the risk of relapse when you combine CSF flow cytometry in CNS leukemia detection. And they are suggested that it may improve future risk stratification, may guide CNS directed therapy. So what we did in Indian children, we wanted to see by combining CNS leukemia detection by combining both flow cytometry and cytologic assessment. So prospectively, we wanted to study. So all newly diagnosed patients at RCC were enrolled after consenting. All patients were treated in the uniform ISICL 14 protocol. So during diagnostic lumbar puncture, 1 to 2 ml of CSF sample was collected. CSF was processed both for cytomorphology and flow cytometry. So both investigations were done by independent investigators. So see, when patients had CNS leukemia, so CSF studies will be repeated weekly till 2 CSF samples are D negative for leukemic involvement. So CSF samples were actually processed immediately, real time. So it will reach the lab, they will process immediately. So for flow cytometry, BNT antibodies, which was a 12-color antibody panel, which was basically used in the diagnosis. A diagnostic marrow sample, what was the antibodies used, was run in the CSF. So presence of leukemic detection was defined as any events which forms cohesive cluster with the immunophenotype, which was similar to the patient's systemic leukemia. So CSF cytology was by the conventional method in the cytospin preparation, and patients were classified as CNS 1, 2, or 3. So our sample size is 325 so far. As of today, we have recruited 87 patients. Majority were BALL, and around 23% of the patients were T. Median age was 4 years. Median WBC count was close to 25,000. We had 1 in 4 patients had hyperleukocytosis, and testicular involvement was 4.5%. So this is the baseline clinical cytogenetic and initial response data of both B and TAL. So TAL, if you see, we have 45% of the patients who had hyperleukocytosis. And so when we compared CSF flow with cytology, so when you combined, so the incidence was actually 29% when we combine both cytology and flow cytometry. But if you see only cytology, 6% of the patient had detectable CSF blast, of which 1 patient was actually flow positive. When we saw, actually they are all reactive lymphocyte. Even flow helps in ruling out a false positive cytology. However, among the flow cytometry positive patients, we had actually 28% of the patient who had flow cytometry positive. So 4 patients had traumatic lumbar puncture during diagnosis. None had cytology detectable blast. However, 3 out of 4 patients had flow cytometry detectable blasts. So median time for the diagnostic lumbar puncture was 6 days. So there was no difference in flow cytometry detectable blast when we do before or after 6 days. However, what we typically note was when we do a diagnostic LP early, the number of events were actually higher. So we can confidently tell whether the CSF is involved or not. So this is the comparison between patients who are CSF negative or CSF was flow positive. So what we are seeing is patients who had a median baseline WBC count higher, and we see higher proportion of patients with high risk having flow positivity. So another thing is response kinetics. When we start administering intrathecal, so after the first IT MTX, still 40% of the patients had detectable blast in the CSF, in the subsequent CSF. However, this is during induction. So after the second intrathecal, 6 patients of this persistent blast, 6 patients cleared. 3 patients cleared after third intrathecal. 1 patient had persistent blood detectable by flow even after completion of induction. So our early study trend shows CSF flow cytometry increases the CNS leukemia detection rates. Even with traumatic LP, flow cytometry can detect leukemia blasts better. So timing of diagnostic LP doesn't seem to affect CNS leukemia detection. Some patients have persistent CSF blast and they have a delayed clearance. So we have to see, means 325 patients data we have to see. So preliminary trend clearly shows a higher sensitivity of CNS leukemia detection by CSF flow. However, we need a longer follow-up in our cohort to assess whether it has a clear impact on the leukemia survival. Also, the response kinetics of these blasts in CSF and its impact will also be assessed as the study progresses. Thank you.
Very good, Dr. Kalashekar. If you can tell me, what were the technical challenges you faced in getting this done?
So basically, we doing a standardization in CNS flow cytometry. Itself, we don't have much literature or guidelines. So we have taken help from ACTRAC team also, from Dr. Prashant Embare and all. So initial 7 patients when we started, actually we didn't have leukemic detection actually. So we have to titrate the procedure, collection, and even the flow cytometry person has to be trained. So what number of events exactly we have to take to have CSF positivity by flow? We don't have a clear guidelines. So we needed support from ACTRAC team also in titrating that.
Kalashekar, you mentioned in the traumatic lumbar punctures that you picked up, like on flow,
Yeah.
you're able to pick up 3 out of 4.
Yeah.
But once it's traumatic, you're going to pick up anyways, because there's
So-
going to be a tiny amount of contamination. Which
No.
maybe you can't see on cytology or cytometry.
Yeah.
Sorry, cytology we
Yeah.
can see on.
So even if you see the literature, even in a traumatic LP, the flow sensitivity is better. So we have a very good literature for that. Even in our cohort also, we are seeing that flow can detect.
No, but what I mean is this a false positive?
No,
How do you know?
No, what we are trying to tell is it may have come from the blood to the CSF, or
Right.
it may be, so actually we are contaminating, it may have a contaminated CSF, or it may be primarily involved. CNS3
But
means
you can't
we
say.
are not able to distinguish. However, you have a CSF which is getting in, having a blast. We don't know through what mechanism it entered, but we will see whether it has a outcome difference as we follow up such patients.
Okay, the second is just a follow-up comment. The Chinese presented data on flow cytometry and CSF for their TLBL study in ASH just last year, a few months ago. So in which they actually omitted radiation. They just gave extra intrathecal.
Yeah.
So, and obviously long-term results again are awaited. We don't know what that impact is going to be. But just like you showed, they cleared the contamination within induction period by the end of 30 days.
Yeah, all these things needs to be followed up, and we are making a small amendment also. Like we want to see the neurocognitive outcomes also in such patients, whether and so we are giving CNS prophylaxis for everyone. Maybe you add a more sensitive technique. Maybe this CNS prophylaxis itself need to be relooked. So all this can be. This study I feel will give a clear clue for the further subsequent studies. Yeah, at the MRD level, madam.
MRD level. We have seen in the bone marrow the hematogons. So is there any equivalence of hematogons in CSF, like they are not basically the active blast, but they have undergone degeneration, but they are picked up as a...
No, madam, we are actually seeing actual blast which are persisting because we are adding the MRD level antibodies, and we are actually comparing it with the baseline positive blast. So we have no doubt in that.
Okay, Chetan's not permitting, so we'll take it up later.
Sorry, we can go to the next speaker, Duhita.
Good afternoon, everyone. My topic for today is trametinib in pediatric low-grade glioma, promises and potential perils. So pediatric low-grade gliomas are nowadays conceptualized as a chronic disease, thereby necessitating multiple lines of therapy. The BRAF MAP kinase alteration is the most common alteration found in pediatric low-grade glioma, thereby necessitating multiple lines of therapy after getting diagnosed. Small molecules targeting this therapy has actually revolutionized the treatment of pediatric low-grade glioma in the last decade. And today we would be sharing our experience of using one such molecule, that is trametinib, which is a MEK inhibitor in pediatric low-grade gliomas, not just as a salvage, but also as a frontline therapy which was used in the last 3 years at our hospital. So coming to the change in approach at Tata Hospital over last 2 decades. So initially it was mainly a radiation-based approach. Then we moved towards radiation delaying and finally radiation avoidance strategies, and thanks to the advent of such targeted molecules. So we have done a retrospective evaluation of all the patients from September 21 to 24 who has received MEK inhibitors, both as salvage as well as frontline therapy. And the decision to start them on MEK inhibitors have been taken on the basis of an MDT decision based on age, clinical and radiological inputs, prior treatments, their molecular status, as well as finances are also taken into consideration. Next, we move on to the methodology. So it was a retrospective audit of all the children who were diagnosed with newly diagnosed or relapsed pediatric low-grade gliomas, diagnosed on the basis of radiology or HPR proven, who has received trametinib as a part of frontline or salvage therapy. We have tried to analyze the efficacy and safety of this treatment, efficacy in the form of overall response rate and safety in the form of toxicity profile, definitely the short-term toxicity profile, and the secondary objective being the clinical profile of our patients along with the short-term survival. The methodologies included our BRAF studies have been analyzed with the help of qualitative real-time PCR. We have used a randomized phase 2 dose of trametinib and dabrafenib, but we do not have the liquid formulations available, so we have rounded it off to the nearest tablet strength. And we have analyzed the response based on the RAPNO criteria 2020 guidelines at 6 months, 12 months, and 24 months into treatment. We have retrospectively collected the data from our electronic medical records and analyzed them in due course of time. So coming to the clinical profile of our patients, the total number of patients enrolled over this period of 3 years is around 32. The median age at which trametinib was initiated is around 7 years, with the maximum number of patients being in the age group of 6 to 12 years. Regarding the modality of diagnosis, the histopathology proven diagnosis was present in about 72% of the patients, and majority of them was pilocytic astrocytoma. As far as the BRAF status is concerned, it was available in near about 78% of the patient, and majority of them were fusion, followed by wild type, and then followed by the mutant ones. Coming to the site of the disease, most of the patients were located in the optic pathway along with chiasm hypothalamic glioma, followed by the posterior fossa, that is cerebellum and brainstem, followed by thalamic. And there were 3 patients who had presented with disseminated disease. Next, we come to, this is a swimmer's plot showing the multiple lines of therapy that was tried prior to initiation of trametinib. So we had given trametinib as a salvage therapy to near about 60% of the patients, and we had also tried giving trametinib as a frontline therapy to near about 40% of the patients. We next move ahead to the response. So the overall response rate of trametinib is around 78%, which comprises complete, partial, minor, stable, as well as major response. And regarding the number of patients continuing with response, at 6 months was around 87%, at 12 months around 80%, and at 24 months around 63%. On the right side of the slide, we have shown the radiological, the MR findings of 3 patients who have more or less showed decent amount of response after initiating trametinib. 6 months, 12 months, and 24 months into the treatment. The first boy was a fusion-positive boy, biopsy-proven pilocytic astrocytoma. We had used it as a frontline therapy because being the location as a brainstem, only a biopsy could be done. And giving trametinib within 12 months of treatment, that showed a decent response to it. The second child, we used it as a salvage because they did not tolerate 2 lines of chemotherapy. And the third, again, had progressed after 2 lines of chemotherapy, was used as a salvage therapy. Next, we move on. So first, it's a waterfall plot showing the overall response to trametinib. There was about 7 patients who had progressed during the course of treatment. And the predominant finding was stable disease, which was fine near about 53% of the patient. And we have also tried to analyze the short-term survival. The 2-year PFS was around 82.5%. We move on to the safety of trametinib. We have analyzed the short-term toxicity profile, and the predominant one was skin and hair toxicity, which was present in near about 60% of the patients, mainly acneiform rash, maculopapular rash, dry skin being the most common, and even paronychia. So there were 8 patients in totality who required temporary drug dose interruption or even requiring drug dose titration, but never required a permanent cessation of treatment. However, there was only one patient who had thrombocytopenia to this extent that we had to stop the treatment 1 year into the therapy. Next, we come to the promises. So definitely it is quite promising, both as a salvage as well as a frontline therapy, and the early response rates are quite encouraging. It is definitely better tolerated than the chemotherapy because there is no need of transfusions, no infections, and no repeated blood tests. We had also tried to circulate a semi-structured proforma to all the patients, but we could gather information from only 14 families. And from that, we have understood that it has a promising role in, as far as school attendance is concerned, reduced number of hospital visits, as well as encouraging their play activities, hobbies, and outdoor activities. Next, we move on to the potential perils. Definitely one of them is the cost of the therapy, which is a major barrier to therapy in resource-constrained settings. The average cost for a 30 kg child is almost around 9 to 12 lakh per annum. The another potential peril is the duration of therapy. None of us knows when to stop and how to stop, whether to taper or titrate the dose or not. And there is another problem regarding the rebound growth that might happen after suddenly stopping the therapy, and what to do after there is a rebound growth. Yes, we have a lot of promising response rates, but most of it is at early, and we need a long-term follow-up of these patients to analyze the long-term survival as well as the toxicity on growth, development, and the endocrine profile. I would also like to conclude that whether trametinib is actually a hope or a hype. So from the small retrospective audit, I think we can possibly safely say that yes, it is feasible to deliver these MEK inhibitors in resource-constrained setting like ours, but definitely funding is an issue. And if generic drug supply is available, that might offset the cost. It has a promising role to delay and also to avoid radiation probably. But we need to go a long way to learn about the optimal treatment duration, the mechanism of resistance in patients who progress or recur. A long-term follow-up is definitely needed to answer some of our questions regarding efficacy, survival, the functional outcomes, and also the long-term toxicity that needs to be analyzed. Thank you.
In your cohort of study patients, what was the median duration of treatment?
Yeah, the median duration was near about 22 months.
You were to stop in
Some patients we have stopped, some patients we are still continuing. So the data is actually too immature to comment anything, but yes, we are also learning as far like how we are administering the drug.
This is a fairly expensive molecule.
Yes.
But you mentioned that about 40% patients were started
As a frontline therapy.
as frontline therapy. How is that decision arrived upon?
Yeah, so these decisions are mainly arrived upon on the basis of our multidisciplinary team meetings. A lot of factors are taken into consideration. Definitely the choice of the family is also one important factor that is taken into consideration. Many of the tumors are not amenable to surgery. There are some patients for whom we did not even have a histopathological evidence of a BRAF status, but still we have started them on therapy and yet to analyze individually all those patients that how they have responded to trametinib.
Right, but just list one or two factors why somebody said, yes, we have to give this MEK inhibitor. We can't give standard therapy.
If you go by the current evidence, it still states that the first line therapy is chemotherapy as far as the BRAF fusion.
I know that, but when 40% patients
Yes.
were started on this,
Yeah.
there must have been some reasons given in these multidisciplinary meetings as to why you were going to opt for this.
Yes.
So just give one or two reasons. Why was this decision arrived upon?
Yeah, so there were two patients with optic pathway gliomas who had deteriorating vision. And since it has quite a lot of promising role that there is no, they can attend their school, the parental unemployment is not uninterrupted. And if the families can afford treatment, so we went ahead and tried giving them the first line therapy.
Thank you.
Yes.
Say, with the deteriorating vision, what is the latent period of after which you start seeing the effect?
So you mean say the median duration of getting the first response? Yes, the median duration of getting the first response in our cohort was near about 11 to 12 months. But the data is too immature to comment upon anything.
Stabilization of the symptoms.
Yes, mainly it is a stable disease, no progression. So in this 32 patients, the small cohort, only one patient has got craniospinal irradiation because he progressed. There was a leptomeningeal dissemination. Only that patient received CSI, but eventually that patient did not respond to that as well.
We now invite Dr. Vandana Dhamankar from ICS to give her presentation.
Good afternoon, everyone. It's a pleasure to be here to present to all of you. My topic is Partnership in Cancer Survivorship Optimization, a comprehensive model for pediatric cancer survivorship care in India. The abbreviation is PICASSO, and I would be talking about the model experience, so you'll be seeing a lot of colorful slides now. So as we all know and we heard in this survivorship track in the EBM, is that survivorship care in India is evolving now and has gained momentum in recent years. There are different care delivery models which are followed in developed countries, but in the country like ours, we are kind of testing different models. So PICASSO project, which is initiated by Indian Cancer Society in May 2016, aims to facilitate the hospitals to deliver consistent and comprehensive pediatric survivorship care through a standardized framework integrating medical and psychosocial support, because most of the time psychosocial support is something which is not really looked into. So this was an important component. The methodology is that once the high volume centers who are interested in starting the clinic approach ICS, we kind of sign an agreement and recruit a psychologist in that unit. They decide the day of the clinic. And if the clinics are already established, then I think it's not very difficult. But where the clinics are not there, the psychologist needs to be recruited and trained in psycho-oncology, post-completion treatment clinics, and then the enrollment starts. So childhood cancer survivors more than 2 years post-treatment and disease-free are registered in these clinics. Late effect surveillance is done by pediatric oncologists, and psychosocial assessments are done by ICS psychologists using standardized and validated tools. ICS also provides financial aid, so the needs which are identified through the assessments, financial aid is provided to the late effect management and rehabilitation support through education scholarships, career counseling, and support groups. This is the spread. So what Dr. Kurkure started way back in 1991 in Tata, and then the UGAM group, which is a childhood cancer support group of Indian Cancer Society, which was started in 2009. This is the genesis of this model. And inspired by this model, now you can see the spread of the Picasso project. There are 16 partner hospitals in 8 states in India. 3 out of these 16 already had the existing clinics, such as TMH, where it originated, RGCI and AIMS Delhi. 3,000 plus survivors are registered in 15 clinics. So this does not include TMH numbers. And the numbers again are, so RGCI and AIMS number would be much higher, but this is, I've only taken when the Picasso project started in that hospital. So what exactly happens in the ACT clinic? When we are saying that pediatric oncologists are assessing, so this is acknowledgement to TMH and Dr. Maya's ACT clinic that the medical proforma is shared, and they kind of assess on the late effects grading and the high risks, all the cumulative doses get entered. On the psychosocial side, it's a screening protocol which is developed. And so all the survivors undergo psychosocial screening as well based on these parameters, such as distress, fatigue, pain, neurocognitive screening. And then those who are kind of having more scores on these screenings undergo detailed evaluation. These are the interventions, psychosocial interventions which are done from the ICS end. And then they are recommended, discussed with the pediatric oncologists, and then further steps are taken. These are again some results to show 3,000 plus survivors are registered. As you see, 690 are from Cancer Institute Adyar. But as I already said, AIMS and RGCI would have much more higher numbers, but this is the Picasso numbers. So now that, it's been 7 years, we tried to do retrospective audit or analysis of a very representative, very small sample from the Mumbai-based ACT clinics, those who were referred to ICS. The focus was again to identify the psychosocial and cognitive concerns and explore relationship with the medical late effects. 102 pediatric cancer survivors data was analyzed. The current age was from 6 to 29, and this was from December 23 to November 2024. The median age was 14.5, and the screening tools, as I said, were the standardized tools which are used in the clinic. 64% were out of Mumbai and 36% were from outside Mumbai. Male to female was 60 to 40. Hematolymphoid was more. What is more interesting to know is that many of them experienced medical late effects, which is 40%. I mean, I wouldn't have the grades as such, but they did experience medical late effects. This is the cognitive data where MMSE was used, and this is courtesy Revati and Linnet, who are our psychologists who have done this study. As you can see, the cognitive, there was no cognitive impairment around 55% of the survivors, but they did have some degree of cognitive impairments. The other survivors did have this. And the interventions used were based on the severity of the cognitive impairments. So those with moderate to severe were also tested for IQ and learning disabilities. That data is being analyzed. This is like Dr. Rachna yesterday mentioned in the panel about the cognitive issues. And, one would wonder that brain tumors would have most, but as you see here, it's a small sample. But as you see here, ALL and even OGS have these kind of issues. There was significant correlation which was found between severity of late effects and distress levels, low MMSE scores, which indicate higher cognitive deficits and higher distress levels. Higher parental distress was also associated with higher distress in survivors. Again, the intervention planned is towards the parents as well. Interestingly, excessive screen time, which was considered as more than 2 hours, was also associated with academic decline. I mean, this is something which is obvious, but this was also the observation in the cohort. Going to the services that are offered by the ICS through financial support, about 450 odd survivors were referred for financial support. As you can see, it is not like one-time financial support that is given. It's year on year that the patients, the survivors are following up, and the psychologists are actually, conducting the performance comparisons. And as you see, around 31% are already improving. The survivors who are enrolled in the scholarship fund are from across India, so you can imagine the challenges that the psychologists are facing. Coming to the last part, which is late effects investigations. This is something which is recently started after identifying the needs, and in 2022 it was started. As you can see, 105 are referred for late effects investigation and treatment fund, majority from Tata Hospital, and other centers are also started referring. So those were, those ACT clinics where the endocrine clinics and the other supportive clinics are already established, those clinics are able to send us these cases. So I must acknowledge the support of the advisors who are advising us on the late effect applications that we get. This is the support group UGAM, as I said, colorful slides. This is something which is very motivating, and it's 15 years old now, and 3 more support groups are already started and 2 are in pipeline. As you see, there is a community app which is started. These are challenges. As you see, sustainable funding, I'm talking from a non-profit angle, has been a concern. I would skip this. So to conclude, the Picasso model has proven to be the replicable model that focuses on holistic care and standardized practices for a lasting survivorship care. And through this project, at least the needs are now identified, and we know that majority of the survivors do require psychosocial counseling as well and support. Some centers such as Chennai and Bangalore have now retained the psychologists. So now they are absorbed on their payrolls. Again, where the proof of the concept is kind of given that for 3 to 4 years ICS has supported, but now these clinics are self-sufficient. They only refer for the rehabilitation. With infrastructure in place, evidence-based insights can now be developed. Integrating, yesterday we heard about the C2S study also. So integrating the ACT clinic concept with the C2S study will ensure the continuity of care. And as I said, it will enhance research, track long-term outcomes, and improve survivorship care models in India. Thank you. Please block the dates. These are international survivors days, which are celebrated on first Sunday of every year. Thank you very much. I would really like to acknowledge TMH being the parent organization, and UGAM, which again motivated this. Thank you.
Thank you. Thank you so much. We have time for only one question.
Thank you, Dr. Vandana. I applaud the effort through the Picasso project, setting up ACT clinics pan-India. It's very difficult. And you have highlighted some of the challenges which you face. Is there any issue with the space constraints, or do you all have a separate OPD dedicated, or is it done within the same, most of the centers, is it done on a similar OPD timing? So does that affect the patient inflow?
Yes, so it's a very important aspect, space and resources. So space is a very important, it's a challenge. And many of the centers like TMH has a dedicated ACT clinic. Chennai also does have a dedicated space for conducting it. But many of the places it is conducted after the OPDs are done. So at least a day is decided where survivors are in advance told about the follow-up date. But space has been a big constraint. Thank you.
And we invite the last speaker, Dr. Aditya Narayan.
Hi, good afternoon, everybody. So I think a lot of us would have seen snapshots of this data in the last 2 days, but I'll be here giving you a little detailed update of the results. So this is the combination of fixed low dose nivolumab and bendamustine in children with high risk relapsed refractory classical Hodgkin's lymphoma. So as we all know, classical Hodgkin's lymphoma responds very well to initial treatment, where right now our survivals have crossed the 90-95% mark, and focus has been on the late effects rather than improvement in survival. But on the other hand, the relapsed refractory Hodgkin lymphoma has we still need to work a lot on the survival, and PFS rates are still between 40 to 50%. This is according to the INFOG recent Hodgkin lymphoma study. The Indian data has showed that relapse rates vary between 10 to 20%. And this is a significant burden that exists in our Hodgkin lymphoma subgroup. So as far as we know, there are no optimal salvage strategies in relapsed refractory Hodgkin lymphoma. There are no clear consensus regarding them. The various strategies adopted, like standard dose chemotherapy, high dose chemotherapy, and now immunotherapy, but there is no clear consensus on which is superior to which. So the response rates to standard dose chemotherapy salvage have varied between 24 to 52%. And apart from the response, it does contribute significantly to the toxicity of the relapse treatment, and it compounds with the toxicity of the transplant, which follows as well. So as we all know, it is really important to achieve a complete remission prior to the autologous transplant. So achieving a good, fast, sustained response in the relapse refractory setting is something very important. So the new drugs which have started to show much promise in this field, both in the relapse refractory and now in the upfront setting, is the immune checkpoint inhibitors, mainly nivolumab and pembrolizumab, and they are being studied. So as we all know, the CR rates of single agent Nivo are low. So it has been shown that nivolumab, now combined with some form of chemotherapy, does produce a good and sustained response. So the something which is very valid and important to our setting is that the standard FDA approved nivolumab doses of 3 mg per kg are the standard doses, but nivolumab in both pharmacokinetic as well as short studies have shown similar promising outcomes at a much lower dose. Both this is in pharmacological studies as well, which show that nivolumab causes a significant amount of PD-1 blockade at very low doses, even as 0.3 mg per kg, which is around 10% of the standard dose. And as we all know, that autologous transplant is still difficult to get in all our patients. So we need to see if there is anything we can move around this. So this is a retrospective study which we conducted across 3 of our Tata Memorial centers from Mumbai, Varanasi, and Mullanpur. And we conducted in all the high-risk relapse refractory Hodgkin lymphoma patients, which mainly included the primary progressive patients, the early relapses, which were less than 12 months, the stage 4 relapses, and the patients who relapsed in the RT field, and 2 or more relapses. So all these patients were given bendamustine at 120 mg per meter square per cycle on day 1 and day 2, and nivolumab at a low fixed dose of 40 mg on day 1 and day 15 per cycle. Response assessment was performed using FDG PET-CT, and complete remission was defined as a DS score of less than 4. And since all of these relapses were high-risk relapse, consolidative therapy with transplant was tried to be done for all patients, and radiotherapy and nivolumab maintenance was offered for selected patients. So the results of the study, there were 18 patients who belonged to this high-risk refractory subgroup. And as you all know, some important findings in this. One, a majority of them were high-risk diseases up front, and radiotherapy was received in around 39% of the patients at initial treatment. And at relapse, we found that majority of the patients had received at least 1 line of therapy, and 25% had received 2. And almost half the patients were progressive refractory disease, which is a very high-risk subgroup. So the important results from this study, the first one being the median dose of nivolumab we used in this study was low. It was 1.2 mg per kg, which is much lower than the standard dose of 3 mg per kg. And among our 18 patients, we noticed that 88%, 16 patients achieved a good complete remission, with all patients being in DS1. And we had 1 patient with no response and 1 patient with progressive disease. Of the 16 patients who achieved a CR, so far 11 have undergone transplant after a median of 3 cycles of this above regimen. And following transplant, 6 patients received no further therapy, 2 have received RT, and 3 of them have been put on nivolumab maintenance. An important point is that when these patients who received nivo-benda were taken up for transplant, the stem cell yield was good, which was previously thought to be a concern because bendamustine is myelosuppressive. The majority of this, overall, this chemo was well tolerated with around 22% patients showing grade 3, grade 4 cytopenias. And we did have one patient who developed a toxic death during cycle 3, during which the child developed pneumonitis followed by ARDS. So in the overall cohort, the median duration of follow-up is still low. We have followed up for around 12 months at present, and the 1-year OS and PFS is 94% and 88%. So overall, this approach of fixed low-dose nivolumab and bendamustine is showing excellent response rate and a very acceptable toxicity profile, and has much promise in our world setting at present. Yeah, thank you.
So only one question. So obviously not all the patients went to autologous transplant, which is a known salvage modality. Am I assuming that's because it relapsed, the staging was different, or was it just lack of resources?
Yeah, this was mainly, I mean, few among the 18 patients so far, we transplanted 11. Among the remaining who didn't achieve transplant, few of them were due to resource constraints, and few of them are yet to be transplanted. So they're just waiting for their slot.
But the plan was to transplant all of them.
Yeah.
And in them, what was the response?
Prior in the relapse refractory setting, none of them received RT prior to the transplant. Only the out of field relapses, the relapses out of field, I think around 2 patients received transplant received radiation post transplant. No, they're less than 15 years.
So while we wait for the judges to finalize the winners, I would like to thank everyone for being here. We approach the end of a very enriching discussion on multiple topics, including precision medicine, survivorship. Today, we also discussed AI, health economics, all of us being Bayesian now onwards. And then we also discussed what is the real world scenario. So that is something that we really enjoyed. I congratulate the entire team headed by Dr. Girish, and whose brainchild this is, and Dr. Maya and Dr. Ram for really taking efforts to really make this conference happen, and also Dr. Gaurav for being the chairperson for the entire conference for this track, along with Dr. Mukta. I thank the administration led by Dr. Sudip Gupta, Dr. Pramesh, for supporting us throughout, and their unwavering support has always been something that we had to make this conference a success. I should congratulate and really thank the CRS team led by Dr. Vikas. It is his first year, and I'm sure he has done a good job, and it will be helpful for him in the future to go along with the success of this conference. Also, I would like to thank Meenakshi and Pratik from the CRS team, who have been instrumental in holding multiple meetings so that we had everything in place for the conference. I would like to thank Amay and Bhanupriya, who have been in the room constantly bearing the cold and making sure that the conference does not go over time, though at multiple times their alarms were ignored. I would thank all the faculty who came in here all the way from different places, and also faculty who came in online at odd hours. It was midnight for them, but they came and they answered our questions. All the consultants and residents from the pediatric hematolymphoid DMGs, the radiation oncology DMGs who came in and supported the speakers. Judges for the conferences. So thank you for everyone for being here. I would like to thank the IT and the engineering team for the audiovisuals and keeping the room cold and comfortable for many and keeping the lights dim so that you don't know who are sleeping and who are not. I'd like to thank the cafeteria team for the excellent food. I'm sure everybody would want the dessert that they had on the first day. They will remember that. Uh, Also the housekeeping for arranging and rearranging the flowers multiple times so that the speakers could be seen. And Dr. Banavali for always being there for us and giving us the reality check as always. And last but not the least, I would like to thank all the delegates from here who joined in person and also a lot of them joining on YouTube live. So thanks everyone, and I hope you have a good day. Now we'll just wait for Dr. Ram to come and announce the prizes. And I would like Dr. Banavali and Dr. Kurkure to come on stage to hand over the prizes.
This is the third pediatric EBM I am witnessing, and it is my fortune. It was first one was 2004, second one was 14 or 15, I don't remember now. And now this is the third one. So I'm sure I'm not going to witness the fourth one because it is going to be a decade later.
A decade, madam. We'll have, don't worry.
So, yeah, I'm just declaring the winners. I'll wait for my turn to say. So I'll be announcing the winners. So we are happy to announce that we had a total of 24 abstracts which were submitted to us, which was quite encouraging and overwhelming for us. And a team of 3 reviewers had judged, and 6 were selected for oral presentation and 12 were selected for the e-posters. So I'll first announce the results for the poster. So the second prize for the poster goes to Dr. Prajakta D. Bhagat from Department of Palliative Care, Tata Memorial Hospital. Her study title was Reducing Procedural Related Pain in Children with Cancer in a Tertiary Cancer Center, a Quality Improvement Project. Dr. Prajakta. Prajakta, congratulations. So she receives a cash prize of worth 2,500 rupees. And the first prize goes to Dr. Niranjan Hegde, Department of Pediatric Oncology, Tata Memorial Hospital. His title is in Survivorship, EOTC QOL for Sexual Health Assessment, Feasibility and Interim Analysis of a Prospective Single Center Observational Study. Dr. Niranjan will also receive a cash prize of rupees 2,500. Moving on to oral presentations, the second prize goes to Dr. Vandana Damankar, title Partnership in Cancer Survivorship Optimization, Picasso, a comprehensive model for pediatric cancer survivorship in India. She'll be receiving a cash prize of rupees 5,000. And the first prize in the oral presentation goes to Dr. Aditya S. Narayan, titled Combination of Fixed Low-Dose Nivolumab and Bendamustine in Children with High-Risk Relapsed Refractory Classical Hodgkin's Lymphoma. Also, he'll be receiving a cash prize worth rupees 5,000. Thank you, sir. Thank you, ma'am. Is it submitted?
Please.